Buenos días a todos, neurocientistas. Un grande gusto para nosotros receberlos todos hoy. Please remember to fill up the questions concerning what you think about the meeting. And also, again, please go to the FALAN platform and give your data in order to have, memory, and in order that Latin America can integrate the neurosciences. Thank you very much for being here today. It's the last day, and stay with us all the time. Thank you. Now, Grace is with you. Thank you.
Good morning, everyone. It is my pleasure to present our speaker. Professor Newton Canteras holds a degree in medicine from Universidade de São Paulo, USP. He also has a PhD in physiology from the same university. He's currently a full professor at Universidade de São Paulo at the anatomy department. And Newton has published more than 100 papers, most of them in the field of neurobasis of motivational behaviors. And just to contextualize to the audience, Brazil has an internal system to grant outstanding scientists with a permanent grant. And Newton is one of the few in the field of neuroscience to hold the maximum position. And recently, I would like to highlight that Newton is a member of the Latin American Research Council, and it is really an honor and a pleasure to have Newton here with us. Let's all come together in thanking Professor Newton for sharing his research with us. Enjoy the lecture, and please register your questions in the chat.
My name is Newton Canteras, and first I'd like to thank the organizers for this nice invitation to give this talk at FALAN meeting. We are going to discuss how the neural circuits mediate innate and contextual fear responses to predatory threats. This presentation will cover from the basic anatomical and functional studies to the more recent chemo and optogenetic investigation of the circuits. First, I will talk about the protocol that we developed to run predatory exposure tests. To achieve a stable and vigorous defensive response to the predator, the animals need to be very well adapted to the environment. So we placed the animals in this apparatus and let the animal in the home cage. At the beginning of the dark period, a door between the home cage and the hallway was open and allowed the animal to explore the rest of the apparatus and obtain food pellets in the food compartment. After an habituation period of 10 days, these animals were very well adapted to the apparatus, and on the 11th day, we placed a live cat in the food compartment. As the animal noticed the presence of the predator, this animal runs away to the home cage and stay there frozen most of the time, presenting a vigorous defensive response. The hypothalamus is the central player for defensive behaviors. The seminal work of Bard and Heisenberger led to the widely accepted view that the hypothalamus plays an especially important role in the expression of defensive behavior. A number of studies have shown that electrical stimulation of the medial zone of the hypothalamus induces a pattern of somatomotor and autonomic responses that resembles the behavior of animals facing natural threats. The same reactions were obtained by microinjections of excitatory amino acids as well as drugs which decrease GABAergic neurotransmission in the medial zone of the hypothalamus. So the medial zone of the hypothalamus is critical for the expression of defensive behavior. Here we have a dorsal view of the medial zone of the hypothalamus from a horizontal section. The medial zone of the hypothalamus is composed by a series of well-defined nuclei arranged from rostral to caudal. Tracing studies using the Phaseolus vulgaris leucoglutamine as anterograde tracer show that the medial zone of the hypothalamus contains two segregated circuits that perform different functions which are critical for the survival of the species or individuals. One circuit is formed by the medial preoptic nucleus, the ventrolateral part of the ventromedial nucleus, the tuberal nucleus, and the ventral premammary nucleus. These sites are sexually dimorphic. They express gonadal steroid hormones and are critical for the expression of reproductive behaviors like mating, maternal behavior, and social aggression. The other circuit is formed by the anterior hypothalamic nucleus, the dorsomedial part of the ventromedial nucleus, and the dorsal premammary nucleus. And by the time we described this circuit, we had no idea about its function. This is the sagittal section of the medial zone of the hypothalamus, shown here, from an animal that received a Phaseolus vulgaris deposit in the anterior hypothalamic nucleus. The Phaseolus vulgaris deposit into the anterior hypothalamic nucleus label the projecting fibers and allow it to visualize the projections fields from the anterior hypothalamic nucleus. In this case, we can see dense projections to the dorsal medial part of the ventromedial nucleus and to dorsal premammary nucleus as well, whereas the fibers tend to avoid other medial hypothalamic sites like the ventral premammary, the ventrolateral ventromedial nucleus, and the medial preoptic nucleus. This circuit is activated in response to cat exposure. Here we show FOS immune-stained frontal sections at the level of the anterior hypothalamic nucleus, ventromedial hypothalamic nucleus, and premammary region. Note that FOS labeling, the expression of FOS protein, is commonly used to indicate the level of neuronal activity, and in this case, cat exposure upregulates FOS expression in the anterior hypothalamic nucleus, dorsal medial part of the ventromedial nucleus, and in the dorsal premammary nucleus. So we have first discovered that cat exposure mobilizes a circuit in the medial zone of the hypothalamus, which we called predatory responsive circuit of the medial zone of the hypothalamus. Predatory cues are processed by different amygdalar paths. We have one path for processing olfactory cues and another path for processing auditory, visual, as well as olfactory cues, and both paths they merge into the dorsomedial part of VMH. Interestingly, the elements of the predatory responsive circuit of the medial zone of the hypothalamus express different kinds of peptides, thus make it possible the use of crylines to target the specific elements of the circuit. The dorsomedial part of the ventromedial nucleus, for instance, express steroid factor 1, and using steroid factor 1 crylines, Diolin's group from University of New York could specifically stimulate the dorsomedial part of the ventromedial nucleus. The optogenetic stimulation of this site provokes freezing, that depends on the projection to the dorsal periaquedutal ray, and also stereotyped escape that depends on the projection to the anterior hypothalamic nucleus. The dorsal premammillary nucleus, in turn, express cholecystokinin, and using CCK Cre lines, a Canteras group from UCLA could specifically stimulate the dorsal premammillary nucleus. The optogenetic stimulation of the dorsal premammillary nucleus produces stereotyped escape that depends on its projection to the dorsal periaquedutal ray. And in this video, we are going to show this kind of behavioral consequences of stimulation of the dorsal premammillary nucleus. Here we have an optogenetic stimulation of the dorsal premammillary nucleus showing escape as well as escape jumps. However, if an escape route is available, the animal that received optogenetic stimulation in the dorsal premammillary nucleus will use this route to escape from the environment, as we can see here. Note that this effect was called context-specific escape and was exclusively found for the dorsal premammillary stimulation and not for other elements of the hypothalamic. Okay. While the stereotyped escape from stimulation of the dorsal premammillary nucleus depends on the projection to the dorsal periaquedutal ray, the context-specific escape does not depend on this projection. Investigating the projections to the dorsal premammillary nucleus, we found that the dorsal premammillary nucleus provides this dense projection to the dorsal periaquedutal ray, as well as a dense projection to the ventral part of the anteromedial thalamic nucleus. And these authors found that this context escape depended on the projection to the anteromedial thalamus. In fact, the anteromedial thalamus is thought to guide this kind of response because this nucleus engages a cortical circuit involved in spatial navigation, which will process the contextual cues to orient the navigation and escape from the environment. And we'll discuss this later. An interesting point about predators is that a good predator never leaves a good impression. And using this same experimental procedure, we're able to see the behaviors of animals exploring the environment previously visited by the predator, which we called an exposure to the hostile environment. In this case, the animal will present a very cautious behavior, presenting these flat back approaches as well as crouch sniffs behaviors. In fact, this animal learned from previous exposure to the predator that the environment is not safe and expresses contextual fear responses, which in this case is characterized mostly by risk assessment responses. Exposure to the context previously visited by a predator activates the same elements of the predator responsive hypothalamic circuit, where we could find upregulation of FOS expression in the anterior hypothalamic nucleus, the dorsal medial part of the ventromedial nucleus, and in the dorsal premammillary nucleus as well. So for the retrieval of fear memory, we engage the predator responsive circuit in the medial zone of the hypothalamus. The question now is, where are the fear memory storages of the predatory threat? It was shown that lesions of the ventral hippocampus and basolateral amygdala could impair contextual fear memory to predators. To test whether the ventral hippocampus and basolateral amygdala could work as memory storages for contextual fear to predator, we injected cyclohexamide, a protein synthesis inhibitor, into the BLA or the ventral hippocampus immediately before cat exposure, which is the acquisition phase of contextual fear responses. Protein synthesis inhibition in the basolateral amygdala, as you can see here, during the acquisition phase impaired contextual fear responses, decreasing risk assessment and increasing time spent in the place visited by the predator zone. Likewise, protein synthesis inhibition in the ventral hippocampus during the acquisition phase also impaired contextual fear responses, as you can see here, decreasing risk assessment and increasing the time spent in the predator zone. Therefore, during the retrieval of fear memory, contextual cues which were previously paired with the predator cues will be noticed by the memory storages in the ventral hippocampal and basolateral amygdala. And as these contextual cues are presented to the animal, these memory storage sites will engage the predator responsive circuit in the medial zone, which through projections to the dorsal periaqueductal gray will influence the expression of contextual fear responses. Now we question how fear memory is encoded. As we mentioned, the dorsal premammillary nucleus projects densely to the ventral part of the anteromedial thalamus. And by the time we planned these experiments, we conceived that the anteromedial thalamus would be involved in the acquisition of contextual fear by engaging cortical circuits likely to encode fear memory. In these experiments, we cannulated the anteromedial thalamus to inject muscimol, a GABA-A receptor agonist, to inactivate the anteromedial thalamus. First, we inactivated the anteromedial thalamus previous to cat exposure. This inactivation did not change the innate defensive responses, but block contextual fear responses. Next, we inactivated the anteromedial thalamus previous to the context exposure, and this inactivation did not change contextual fear responses. In this way, we came up to the conclusion that the anteromedial thalamic nucleus influences the acquisition but not the expression of fear memory. We next investigated the cortical projections of the ventral part of the anteromedial nucleus and used the phasellus vulgaris as an enteroid tracer. This is a phasellus vulgaris injection into the ventral part of the anteromedial nucleus. And here we show the cortical projections of the ventral part of the anteromedial thalamus, and we can see dense projections to the prelimbic and anterior cingulate area, as well as in the visual association area. This is a flat map representation of the cerebral cortex, where we have the main cortical fields that receive projection from the anteromedial thalamus. And these fields, together with the retrosplenial cortex, they form a cortical circuit where the anterior cingulate area occupies the center position. Elements of the circuit are especially active or responsive during cat exposure. We have shown that cat exposure produces a much larger increase in FOS expression in these cortical areas compared to adjacent cortical fields, suggesting that this cortical circuit is particularly responsive to predatory threat. The circuit responsive to predatory threat projects to the hippocampal formation, mostly to the parahippocampal region, and to the amygdala, forming a complex cortical hippocampal amygdalar circuit which should guide spatial navigation during the context-dependent escape, as previously explained. And as we shall explore, this circuit is also involved in fear learning through predatory threats. In the studies that we explore how the cortical circuits influence the acquisition and expression of contextual fear, we changed from rats to mice and use this paradigm. The predator exposure test. In this paradigm, we took a mouse and placed in the apparatus together with a live cat. As the mouse noticed the cat, this mouse flew away to the box 1, where it remains frozen and risk assessing. During the context exposure, the apparatus was cleaned, and the animal will explore the apparatus, presenting these flat back approaches, and avoid the box where we kept the live cat, and tend to stay in box 1, risk assessing the environment. As we mentioned, the anterior cingulate area occupies a central position in the predatory responsive cortical circuit. Here we examined how this cortical field influences the acquisition of contextual fear. To this end, we inactivated the anterior cingulate area and showed how this inactivation interferes with fear memory. This inactivation was carried on using inhibitory dreads, which is the acronym for designer's receptor exclusively activated by designer drugs. We bilaterally injected the anterior cingulate area with a adeno-associated virus coding for inhibitory dread, as you can show here, fused to a fluorescence protein, in this case MC3. Here we show the transfected cells in the anterior cingulate area, which appear fluorescent and express the inhibitory dread. Clozapine N oxide CNO was injected systemically to link in the inhibitory dread and promote the inhibition of the anterior cingulate area. Inhibition of the anterior cingulate area during the acquisition phase of the contextual fear does not change innate responses, but impairs contextual fear responses. Thus, we are able to see in those animals that express this inhibitory dread, that these animals decrease risk assessment and increase exploration when exposed to the context. Thus, we are able to see that the anterior cingulate area is involved in the acquisition of contextual fear. In the following experiments, we investigated which sources of inputs to the anterior cingulate area are particularly activated during the acquisition phase of contextual fear memory. First, we injected fluorogold, which is a retrograde tracer, into the anterior cingulate area. Fluorogold was injected to label the afferent sources of input. The animal was left during 7 days to allow the fluorogold retrograde transport and retrogradely label the afferent sources of inputs, as we can see here. After 7 days, this animal were exposed to a cat, and 9 minutes later, these animals were perfused to examine FOS expression in each afferent source. So, we found a particular number of FOS fluorogold double-labeled cells, in other words, cells that were activated in the thalamus, in the central lateral thalamus and anteromedial thalamus, as well as in the elements of the cortical circuit responsive to the predator in the visual association and retrosplenial areas. Next, we silenced the projection from the anteromedial thalamus to the cingulate region to investigate how it influences the acquisition of contextual fear memory. To this end, we use optogenetic silencing. We use adeno-associated virus coding for allorodopsin fused with a fluorescence protein. Transfected cells, they express the fluorescence protein together with allorodopsin. And photostimulation with yellow laser in the axon's terminal will activate the allorodopsin, which is a chloride pump that hyperpolarizes the axon, silencing the projecting fibers. We injected adeno-associated virus coding for allorodopsin in the anteromedial thalamus and placed the optic fibers in the projecting field of the anterior cingulate area. Silencing the anteromedial path to the anterior cingulate gyrus during cat exposure, as you can see here, did not influence innate behavioral responses, but impair the contextual fear responses, as we can see here, decreasing risk assessment and increasing fearless exploration. Thus, the inhibition of the path from the ventral anteromedial thalamus to the anterior cingulate area impairs the acquisition of contextual fear memory. We next investigate how the anterior cingulate area would be able to influence the acquisition of fear memory. And to understand how that would be possible, we took Guilmartin hypothesis, where the anterior cingulate area is likely to influence the memory storages in the amygdala and the hippocampus through projections to the amygdala and to the rhinal cortex. And we try to prove this hypothesis. First, we examined the projections of the anterior cingulate area. A adeno-associated virus coding for fluorescence protein was injected into the anterior cingulate area to label the projection fields. And we can see that the anterior cingulate area provides dense projections to the basolateral amygdala and the perirhinal cortex, which would be the targets involved in the acquisition of fear memory, and also projects densely to the dorsoparacrital gray and postsubiculum. Here we use the optogenetic inhibition previously described to silence the projection from the anterior cingulate area to the basolateral amygdala. Silencing this projection during cat exposure did not affect innate responses, but impairs contextual field responses. However, silencing this projection during context exposure did not change the contextual responses. Therefore, this path is involved in the acquisition, but not the expression of contextual field responses. To see the level of activation of the paths, of this particular path, during the acquisition and during the expression of contextual field responses, we use a functional tracing. We injected fluoro gold in the basolateral amygdala. Fluoro gold is a retrograde tracer, and we try to label the projection from the anterior cingulate area to the basolateral amygdala, and the injection of fluoro gold here will label the cells in the anterior cingulate area as we show here. We left 7 days to allow the retrograde transport of fluoro gold. To see the false response to the predatory response, in this case, animals were exposed to a cat and perfused 9 minutes later. Then we were able to determine the percentage of this fluoro gold labeled cells that also express FOS. In other words, the cells that project to the basolateral amygdala, which were activated during cat exposure. And we saw close to 80% of cells presenting double label fluoro gold and FOS, showing that this path is highly active during the acquisition period of field responses. Running the same functional tracing strategy during the expression of contextual field responses, we saw only a small number of FOS fluoro gold double labeled cells, suggesting a weak activation of this path during the expression of contextual field. We repeated exactly the same strategy for the projection from the anterior cingulate area to the perirhinal cortex. We silenced this projection during exposure to the predator, and when we did that, we did not change the innate fear responses, but impair the contextual responses. When this projections is silenced during the context, it doesn't affect the contextual fear responses, showing that this projection is also involved in the acquisition, but not the expression of contextual fear responses. Running the functional tracing, we saw that during the acquisition, this path is active, is very active. 50% of the cells double-labeled phosphorus and fluorogold. However, during the expression, only a small percentage, like 50%, presented this double-labeled phosphorus and fluorogold. Next, we examined another important target of the anterior cingulate area, the postsubiculum, and silence this projection. Silencing this projection during exposure of the cat did not affect the acquisition of few responses, and silencing this projection during the context also did not affect the contextual few responses. And the functional tracing also showed that this path is weakly immobilized during the acquisition as well as in the expression of contextual few responses. Therefore, we were able to confirm Martin's hypothesis and showed that the anterior cingulate area influences the fear memory acquisition through projections to the amygdala, the basolateral amygdala, and the perirhinal cortex, thus influencing the memory storage sites in the basolateral amygdala and ventral hippocampus. Apart from being involved in the acquisition of contextual fear responses, our inactivation also showed that chemogenetic inactivation of the anterior cingulate area during exposure to the context also impairs contextual fear responses, and therefore is also involved in the expression of contextual fear memory. We saw that the other targets examined so far did not influence the expression of contextual fear responses. Another major target of the anterior cingulate area is the dorsolateral periaqueductal gray, as you can see here. And in those experiments, we inhibit this path with the same optogenetic silencing strategies. Inhibition of this path during cat exposure did not affect innate or contextual fear responses. However, inhibition of this path during the context impairs contextual fear responses. Functional tracing indicates that this path is weakly activated during the acquisition of contextual fear responses, but is largely activated during the expression of contextual fear responses. Therefore, the anterior cingulate targets are differentially recruited during different phases of the aversive memory processing. The projections to the basolateral amygdala and perirhinal cortex are critical for the acquisition of contextual fear memory, whereas the projection to the dorsolateral PG is involved in the expression of contextual fear memory. To see the distribution of projecting cells to the anterior cingulate area, to see in the anterior cingulate area where the cells are to project to the basolateral, periaqueductal gray, and perirhinal cortex, we run experiments using 3 different retrograde tracers in the same animal. They received fluorogold in the basolateral amygdala, a retrograde track tracing virus in the periaqueductal gray, and cholera toxin in the perirhinal cortex, and labeled in the same animal the cells in the anterior cingulate area projecting to basolateral amygdala, projecting to periaqueductal gray, and projecting to perirhinal area. Merging these images, it's clear that the projections to the sites involved in the acquisition of fear memory, the basolateral amygdala and the perirhinal cortex, they are located in the supragranular layers, layer 2 and 3, and show 50% of double labeling, suggesting that there is a large proportion of cells in the anterior cingulate area that projects to both the basolateral amygdala and perirhinal cortex. In contrast, the projection to the periaqueductal gray arises from the infragranular layers and do not overlap with the projections to the BLA or the perirhinal area. To summarize the findings, we have that predator cues are processed by amygdala PETs to inform the medial hypothalamic circuit responsive to predators, formed by dorsomedial VMH, dorsopulmonary, and anterior hypothalamic nucleus to inform about the predator presence. The stimulation of each one of these elements produces a particular type of defensible response. We showed here that stimulation of the dorsal medial part of the ventral medial nucleus produces freezing that depends on the projection to the dorsal periaqueductal gray, and stereotyped escape that depends on the projection to the anterior hypothalamic nucleus. The anterior hypothalamic nucleus, as we have shown in the beginning of this talk, projects heavily to the dorsal premammillary nucleus. Stimulation of the dorsal premammillary nucleus produces stereotyped escape and a more elaborate form of escape, the context-specific escape, that depends on projections to the anteromedial thalamus, which engage cortical navigation circuits to guide the escape responses. The encoding of fear memory also depends on the projection to the anteromedial thalamus, which engage cortical circuits related to fear learning. In particular, we showed that the projection to the anterior cingulate area is critical for the acquisition of contextual fear memory. The anterior cingulate area, in turn, is likely to influence memory storages in the basolateral amygdala and ventral hippocampus. Finally, the ventral hippocampus and basolateral amygdala may be viewed as fear memory storages to predatory threats. And as contextual cues are present to the animals, the memory storage sites, the ventral hippocampus and basolateral amygdala, engage the predator responsive circuit in the medial zone of the hypothalamus, which through projections to the dorsal periaqueductal gray influences the expression of contextual fear to predator threat. Note that we are also showing the path from the central amygdalar nucleus to the ventrolateral periaqueductal gray, a pathway largely explored in fear conditioning studies. However, this path does not work for fear conditioning to predatory threats. Importantly, central amygdalar nucleus lesions do not affect contextual fear to predatory threat. In this way, it's important to note that studies using fear conditioning to painful stimuli do not predict the neurosystems underlying contextual fear to predatory threats. Finally, I would like to acknowledge my collaborators who help put together the story. I start with Larry Swanson, with whom I work to trace the hypothalamic circuits. Caroline and Robert Blanchard, and they provide the basis for our behavior analysis. Dr. Abhishek Adhikari, who performed the optogenetic experiments showing the context-dependent escape in the dorsal premammary nucleus, and my collaborators at the University of São Paulo, in special Dr. Miguel Xavier de Lima, who performs the studies on thalamocortical circuits related to fear memory. Finally, I would like to acknowledge the funding agents, CNPq and FAPESP. Thanks.
So, first of all, thank you, Newton, very much. Let me mute the video. You probably are listening echo. Now I think it's fine. Thank you very much for the excellent talk. It's really nice to see how the modern neuroanatomy works and how to do it very well. So exploring all the pathways and like you chose the paradigm, a behavioral paradigm with a very reproducible and strong behavior, and then you move towards the neurocircuits that fundament this behavior. So I have some questions. One is more methodological. So do you still use a real cat to run this paradigm?
Yes, a real cat. There is nothing that can substitute or replace the real cat for the experiment. And the experiment is not that bad because we get like a house cat, and we just place the cat in the together with the rat or the mouse, and we are using mice now. The apparatus, and they stay well. There is no problem. Obviously, the rodent doesn't stay well, but for the cat, there is no problem. There is no problem. And still, and this is very important, because to get good contextual responses, you need a cat. Sometimes you can run the same experiments using a rat. It's the rat exposure test, but rats, they don't yield a very good contextual response. So the good contextual response you get from cats.
I'm asking because we try in our lab to run an experiment using cats odor, and for some reason, it's not the same paradigm. It's just it's more related to olfaction. But the mice, they did not fear the cat's smell. So that's why I'm asking.
You need to be cautious. We did in fact, we ran cat odor, cat exposure, rat exposure in this Adhikari's work in the dorsal premam. We could show that the dorsal premam is responsive to several kinds of threats, including odor. And this odor tests, they can be very tricky to test. So you need to be sure that you get a strong cat odor in your sample, and sometimes it's not easy to get. There are some tips that we can give you then to get this thing. But I do prefer to work with live cats, and responses far more robust than other threats, minor threats.
Great. So another question is about the duration of this contextual memory, how long it lasts, because you test 24 hours later, but have you tried to see how long it lasts?
We tried in rats. Rats, they have a more strong expression of contextual memory, and we tried that for it lasts at least for 1 week, and after and then of course you start getting a extinction of this memory, and curiously after 10 days or 11 days or something, you get a spontaneous renewal of that memory. So it is a typical extinction process. There is extinction process in this.
Okay, so we have a question about other predators, smell, like, or not smell, but predators. Have you tried, or what do you think about snakes, for example? It should be, did you expect the same pathway involved?
No, actually, we just submitted a paper for snake threats. We tried the snake threats as well. We did one paper first with Norberto at Ribeirão Preto, and then we decided to run an experiment where we could separate the snake smell and the snake presence. And then we have a very interesting, because when you have odors from mammals, carnivores, which are you have activation of the dorsal medial part of the ventromedial nucleus, and they test several kinds of different odors, not just cat odors, several other kinds. And then when you have, let's say, exposure to the snake skin, you have activation of another path. So the response to different predators, they are different. What I talked today is just mammals, carnivores, okay? But if you're talking about the snakes, you have a different different system. You have some overlap, but respond a little bit differently in terms of the smell. The odor path is different. It's very interesting. You have a different kind of predator being processed by different parts of the ventromedial nucleus. Is not so much the dorsal medial, but the central part of the ventromedial nucleus. And from there, all the tracing and all the circuits they are a little bit different.
Very interesting. We have another question here from Carlos Humberto Andrade Moraes. He said, I was wondering if the dopaminergic mesolimbic pathway and the nucleus accumbens could somehow be involved in the behavioral responses in contextual fear.
Yes, in this case, there is one important path of this, important part of this path that I did not explore, and I think it's related to that, is related to the accumbens and the modulation of the accumbens by the basolateral amygdala. And of course, this mesolimbic should be involved in this part as well. So, so far, we did not explore this. This is one of our next goals, to explore the basolateral amygdala and the accumbens relationship in terms of anti-predatory responses. Nobody has, we noticed that if you lesion the basolateral, you have changes in the memory, you have changes in the innate responses, but we don't know how it works. And then you bring the accumbens to this circuit. So far, we didn't bring the accumbens to the circuit. The accumbens is, the nucleus accumbens is there, and we need to bring the accumbens to this circuit and to put another layer of complexity to the circuit.
Very nice. I was wondering, so how plastic this kind of memory is? For example, have you tried or have any thoughts about how the interoceptive context of the animal influences in this behavior and activation pathway? Let's say now we are living in a complex, different context, like worldwide, this pandemic, and maybe the fear is not it's not like the same. We have different threats. I was wondering if this very innate fear pathway could be changed if the animal is like with stress or something like that.
Of course, you can modulate the response. That's for sure. This is one thing. I I don't have an answer for your question. Of course, I don't. But I can tell you what. Many researchers, they complain that the response to the predator when they are running these tests, they are not, let's say, stable. And if you take a rat, for instance, and put together with a cat, sometimes you get a response, sometimes you don't. And it's very important to get a stable response to stabilize the test. So from there, you can see that this innate response is not so so robust every time they put together. So to get a stable response, the animals need to be very well adapted to the environment. If they are not, you don't get that. So this is why we habituate the animals for a long period. When we start running these experiments, we had this problem. We have a very a variation in terms of response very large. And then when we start doing this long habituation periods, we start getting a very stable and robust response all the time. So in other words, this response is modulated by many factors. It's not, it's there, but there is a strong modulation of several factors.
Mm-hmm. Yeah. No, I'm asking you that because usually I have a feeling that innate behavior are very strong and not plastic at all, but
no, they are. It can have a lot of modulation, and that's for everything, for eating behavior, mm-hmm. for mating response, for social aggression, for maternal aggression, for everything. You can have different kinds of modulation, different kinds of response.
So we still have time for more questions. I have one more at least. Mm-hmm. So I have a lot. I would stay here forever with you discussing this topic. It's very interesting. But I was wondering if that you actually saw the pathway from the anterior cingulate cortex to basolateral amygdala, and then, if I'm not wrong, the periaquedutal gray. Yeah. Mm-hmm. Have you seen like bottom-up projections, like, or that in somehow the most caudal areas connect and communicate with rostral areas? Like from black to anterior cingulate?
I see. No, this is why we run these experiments using fluorogold. And and this is interesting. During acquisition, we place fluorogold, and then we're able to see all these up projections that I mentioned, all those projections that are affecting the anterior cingulate area during the acquisition phase. So during the acquisition phase, we have you can see that you have this path from the thalamus that is affecting, and this is going to be bring together with the context, and the context is being processed by the retrosplenial and the visual association area, and they go together in the anterior cingulate area. So you have in the anterior cingulate area the register of the context and register of the predator. Both are there, and then those things go to the projection paths. The basolateral amygdala doesn't go; it goes back a little bit to the anterior cingulate area, but not active during the process of neither the expression or the process of acquisition. We saw that. We also we did similar things we did for the acquisition, also for expression. And for the expression, we have not many places that that you can see that active during the expression. We couldn't figure out what's going on there. So you have back pathways, but the most interesting that we could see so far are the ones using for the acquisition phase, and that we can see this clear computation of the anterior cingulate area related to the predatory threat and the contextual cues.
Very nice. We have a lot of people congratulating you, saying that the talk was really nice, and Norberto Cairasco is saying that you could use his cat, Kekeiston.
It's a no joke. It's a no joke. I know what he means. It's a no joke.
Okay, and I think that's what we have. I don't know how many time we have left. If there's no further questions, I would like to congratulate you again, Newton, and thank you for the excellent talk. And that's it. I think that we have to finish. Thank you very much.
Thank you very much. Thank you for the invitation. It was an honor to be here, and thanks.
Okay. So good morning, everybody. My name is Francesco Rossi from Uruguay, and today I'm very happy, pleased, and honored to chair the following symposium that is entitled Neuroscience and Education. First of all, I would like to thank the speakers that kindly accepted our invitation to contribute to this very relevant topic for our region. So today we will have Roberto Lent, who is Emeritus Professor at the Universidade Federal do Rio de Janeiro, Senior Researcher at the Dor Institute of Research and Education, and General Director of the Brazilian Network of Science for Education, with his talk, Neuroscience for Education, the Start of a New Era. The second speaker is Dr. Andrea Paula Goldin from the Universidad Torquato di Tella and CONICET of Argentina, with the talk, How can cognitive neurosciences contribute to education? A case report. Then Professor Juan Carlos Valle Lisboa of the Facultad de Ciencias and Centro Interdisciplinario de la Cognición para la Enseñanza y el Aprendizaje of the Universidad de la República de Montevideo, Uruguay, who will present his talk, Cognitive Science is the Bridge between Neuroscience and Education. And finally, we have Professor Norberto Garcia-Cairasco of the Ribeirão Preto School of Medicine of the University of São Paulo, Brazil, with the talk Building Citizenship with the Network Science, Arts, Education and Society, ScienArtES. So before beginning, I would like to remind you that if you want to ask questions, you can exclusively use the chat through the FALAN platform or the YouTube channel. We will have a short time at the end of each presentation for a rapid question and a 25 minutes time at the end of the symposium for more questions and discussion. So thank you very much for your participation. I hope you enjoy the symposium. There is also our colleague Maria Eugenia Pedreira, who will help us with the chat, the question in the chat. So we can begin with our first talk of Professor Roberto Lent. Thank you.
Hello, good morning. Thank you very much for the invitation to speak at this FALAN meeting. I will share my screen so that you can follow my slides. One moment, please. Okay. I guess you can see my slides. What I will speak about today is explore a little bit the concept of science for education, which is a new concept that is expanding its influence in different countries, in different regions, as well as in Brazil. In particular, I will give examples of neuroscience because it's my field of work, but as you will see, the concept is broader than neuroscience. The concept is based on an idea by Donald Stokes, proposed in 1997 in this book that you see here, which is the Pasteur's quadrant. So the Pasteur's quadrant is this one here in red, and it means research inspired by use. This kind of research, as that performed by Louis Pasteur, responds, answers two very important questions. One here, fundamental concepts, does it generate fundamental concepts? And the second here, does it have any practical use? So as you know, Pasteur has defeated the Aristotelic view that life can be generated spontaneously, which is a fundamental concept in biology. And he also has developed technologies that are still in use, such as the pasteurization of foods and drinks and wine and milk, etc., the pasteurization of foods. Okay, the Pasteur's quadrant is a kind of translational research, and it has been applied very successfully in health. It is described by this saying, from bench to bedside, whose players are very well known, universities at one side, health systems at the other side, and different kinds of business and intermediaries here making the bridge between the two sides. Now, what is the scenario that resulted from the employment of this concept of translational research in health? In Brazil, child mortality dropped very pronouncedly in the last decades, as well as life expectancy has increased a lot, changing the demographic pyramids, as you see here below. Now, we don't know what's the impact of COVID-19 on these curves, but I believe that after the pandemic is controlled and transformed into an endemic, or perhaps an epidemic, we will return to these curves as translational research continued to be applied in health in most countries of the world. Now, what happened to education? Education can be measured by different scores, different assessments. One of them is the PISA score. You see here all the countries aligned according to their results. The average is pointed in the middle, and the position of Brazil is very bad. We are in the 65th position in the world. Now, if we take this performance along time, along the at least the two last decades, we can see below in blue that Brazil has perhaps a subtle increase, a subtle improvement, approximating the average of OECD countries. But when should we reach the average? We can project this curve to the future, and we will arrive a confluence of the two curves perhaps in 2060, and we cannot wait so long to have results. So the hypothesis we work with is that, of course, we don't know whether how the coronavirus, the COVID-19 pandemic will impact these curves probably makes things even more difficult. But of course, the hypothesis that we examine, we adopt is that perhaps science, the inclusion of science in the form of translational research to education will allow an increase in the derivative of this curve in Brazil, as well as in other countries, and allow a more efficient result in the end. Now, how would it work? Is it possible? The architecture will be the same from bench to the classroom. The players would be similar, except that educational systems at the right would substitute health systems, as I showed previously, and you would have different intermediaries at the middle. Now, how would it work? Actually, it somehow works, but in a very ineffective way. Translational research in education could be reactive as the first format, by which interventions are proposed without scientific evidence. Many different laws, rules, etc., are made by different governments. They are implemented by the educational systems, and they are assessed by a rigorous methodology. We have many in Brazil, and there are many in different countries. Now, this model brings a higher risk of failure because the intervention is not based on science. It is intuitive, sometimes ideological, some other times, and it has to wait 10 years to be assessed. Now, what we propose is that the model could be proactive. That is, the intervention would be based on science, on scientific evidence. So the governments would ask scientists about the underpinnings of learning, the influence of different factors, different environmental factors on neuroplasticity of the brain, for instance, and many other issues. And if intervention itself is based on scientific evidence from the beginning, the risk of failure is lower because things could be corrected in shorter periods of time. Now, based on these kind of concepts, we founded in 2014 the National Network of Science for Education, of which I am the Director General at this moment. And our mantra is an attempt to convince social players to foster translational research inspired by the problems of education. So it would be the quadrant, the Pasteur's quadrant applied to education, which means to create an ecosystem in the country by which there would be a confluence of disciplines directed to education. So this idea is, it doesn't exist at this point, but we propose that it should be adopted by our country and perhaps different other countries. Neuroscience has a role in particular, and I will give you an example, a very humble example, but very nice, very important. It's a work made by João Ricardo Sato and Guilherme Brockington from ABC University in São Paulo. They employed NIRS, which is a technology that very friendly and not very expensive, that allows recording the brain of different subjects at the same time. It is better in some respects compared to electroencephalogram, and similar to functional MRI, with some advantages that I don't have time to explore right now. So the example is the following. The example that I chose to show you is the following. The set is a child being exposed to a class given by a teacher who conveys information in the board and speaks and talks viewed in the presence of the boy. The brain of the boy is recorded with NIRS, and simultaneously his eye movements are recorded with an eye tracking system. So we have access to both brain waves or brain activities in different regions, and to the focus of attention of this boy when being exposed to the class given by the teacher. So I'll show a very short video in the next slide, and you should pay attention to the green circle that is shown here at the bottom right. It will demonstrate, it will show the focus, the variations of and the position of the focus of attention on the eyes of the teacher. You arrive at the conclusion that they are focused about 50% in the two different places. So the kid is interested in the information that is conveyed on the board, but he's also interested on the eyes of the teacher. And what do the eyes of the teachers convey? Empathy. So empathy is a very important element of teaching and learning. And this raises a number of different issues that can be explored in the discussion. That is, remote teaching eliminates empathy because the teacher is speaking in the, as here, for instance, all the viewers are hidden using the Zoom platform or whatever platform. So there's no face-to-face exchanges between students and teachers. So this is, of course, one single very simple example of one kind of research that can be made to inform about the process of learning, the processes, the mechanisms of learning that can illuminate the measures, the interventions proposed by educational policymakers. Now, to finish, I will mention a project that we have to give a broader and an even broader range to this effort in favor of science for education. That is the proposal of creation of a UNESCO chair on science for education. Some of the proponents are present in this table here. And if we succeed with the UNESCO evaluation of the proposal, we will be able to give an international dimension to this kind of network. Of course, we have other, there are other initiatives in this same track that we can discuss afterwards. So thank you very much. I will end by calling the attention to the importance of building this bridge between science and education. It's not impossible, it's not too far, it's very feasible, and it depends on the strategy we use to do it. So thank you very much. And I'll stop here. And I'm again, thank you, and I'll be.
Well, there are so many questions that we can ask. I was curious, let's say personally curious about the magic word empathy, which is another entire word, I suppose. And but can you just tell us an example of how empathy can be translated to policy? I mean, I have a general idea, but what can you tell us about this?
Well, that's a very difficult question. Empathy, of course, is first of all difficult to define, and second, it's difficult to measure objectively with scientific tools. What we can say is that empathy requires face-to-face interactions between students and teachers. And particularly now that we face the pandemic and we are forced to employ remote tools to teach and the students to learn, empathy is highly impacted. But we don't have the measures to evaluate how deep was this impact. Of course, it's obvious that a remote interaction between students and teachers reduces the possibility of empathy, and we will probably have to find ways to deal with it after the pandemic, because the remote tools will remain, but they will have to be balanced with face-to-face interactions to guarantee that empathy is used in the learning process.
Yes, it is really a difficult question on how to measure empathy and translate it. So we will see. So thank you very much, Roberto, and I will have a more general discussion at the end of the symposium with other questions. And we can go now to the second speaker, Andrea Goldin, with her talk on how cognitive neurosciences contribute to education, a case report. Thank you.
So, hi, thanks Francesco and the Sociedad Argentina de Investigación en Neurosciences and the FALAN authorities for inviting me to this symposium on neuroscience and education. So let's start with the brain. So with the brain, we interact with the world, and we can respond to that information, and the responses might be chemical or physical or behavioral, and these responses might as well change the brain itself. So this plasticity modification implies that even though the information might be the same, the brain is different every time and understands different. And this is what we call learning, the learning process. And the other side is also that also is part of education, is the teaching process. And we are not going to talk about teaching today, so let's focus on this plasticity feedback loop, which implies that in order to learn something, the brain has to be prepared to learn it. For instance, in order to understand Math 3, first, hopefully, you should understand Math 1, and before that Math 0, and before that you should have to understand basic math operations. So the brain, this implies that the brain has to have the proper capacities already built in order to learn something else. So you can use in one situation what you learned in a different situation. And so this it's been known for way more than a century, and it's called transfer, and it's the influence of the improvement in one mental function upon the efficiency of other functions, or the influence of the decrement in one mental function upon the inefficiency of other functions, or the efficiency of other functions. So this that might be so clear with Math 1 or 3 is not that linear with other cognitive capabilities. I'm specifically thinking about some cognitive processes that are also associated with the frontal lobes and that regulate and coordinate other processes, perceptual, motor, or emotional processes. And these are called executive functions. And these executive functions are, there are 3 basic executive functions: working memory, inhibitory control, and cognitive flexibility. And there are other superior executive functions, but altogether, by using them, we can, they allow us to do or to learn or to socialize as much as we want or as much as we can. By using all of them, we have a proper interaction and a proper learning from the world. And that's why some authors like to call them the foundations of cognitions, because those are cognitive capabilities over which all the other aspects of cognition are built. And so by using all of them, you can, for instance, pose an objective and plan how to achieve that objective and stick to that plan. And if something changes, they allow us to be flexible enough as to change the plan and eventually even set new goals. So we are really using executive functions not only to play or to socialize, but also to learn math and even to understand this talk that we are having here. We do know that better executive functions are associated with better outcomes of life in general. For instance, better executive functions are associated with better academic performance, with lower body mass index, with better jobs later in life, with less probability of developing an addiction. So with this in mind, over a decade ago, we started to develop Mate-Marote. Mate-Marote is a software that stimulates executive functions. Then this is called cognitive training software. And it's specifically tailored for children between 4 and 8-year-olds. Mate-Marote is free and is available online. And basically what we do is, through game, we stimulate these executive functions. And basically what in a class, some children play the games of Mate-Marote, other children play other games that are equally motivating, but don't train executive functions that much. And to assess whether the games actually trained or not executive functions, we test children before and after the interventions, and we compare the pre-test, post-test. And after doing this for several years and different experiments in different with children from different backgrounds, we learned that the games actually trained these executive functions. But what we thought is that, okay, so it's very important that the games train executive functions. We want that, we need that, so because better executive functions are associated with better whatever later on. but we wanted to see something. And so to see something in real life. So if executive functions are actually used for anything that we do, then we should find something from real life that would show the changes made by Mate Marote. And we thought, okay, so these are 6, 7, 8-year-olds. So what's important for them and for their communities is that, okay, they learn at school, that they make it to the following grade. And so we went to look for the grades of the students. Grades were given by the teachers of the children, teachers who are completely blind to the experimental hypothesis. They didn't even know that there was a control group. Children, of course, didn't know it either. And this experiment I'm going to show you was done with first graders, so 6 to 7 years old, over 100 children in the city of Buenos Aires. Grades are given every 2 months at the end of each bimester. The intervention took place between the 3rd and the 4th bimester. And so we consider grades given in the 2nd bimester as the pre-intervention grades, and grades given in the 4th bimester as the post-intervention grades. And what we found is basically that at the end of the school year, grades are higher than at the beginning, but no differences were found between both groups. And this is important because those children that were trained, that played with Mate Marote games, actually improved their executive functions, but that improvement wasn't reflected in the grades. But I didn't tell you this is just a subgroup of the children. I didn't tell you that if you take a group of students and you can arrange them by the attendance to school, and what you would find is that there is a correlation between the rate of attendance and the grades given to that children, so given to that students, as that better, more attendance correlates to better grades. And what I showed you here was if you split children by the median of attendance, you have the high attendance and the low attendance group. And what I showed you here correspond to the high attendance group. When you go and see what happens with the low attendance group, a similar pattern is seen, but very different. What you find is that those children that played with Mate Marote improved much more their grades than their peers that played with the control games. And this improved so much as are indistinguishable from their peers who had the higher attendance. And this is very important because these are 6-year-olds. So if they don't attend to school, it's because there are social and familiar circumstances that doesn't allow the children to attend so often school. And what we found is simply that by boosting their executive functions, their brains are more prepared to learn wherever their teachers were going to teach them in language and math in first grade. So where are we going? We're doing a lot of collaborations. We work very closely with a group of Uruguay, and we are now starting a collaboration with a group of the Universidad de Barcelona in Spain. We are open for collaborations. We are also starting to work with elderly people. We are trying to apply Mate Marote in order to change the cognitive decline of the elderly, but this is for another talk. We do all this work with the Applied Artificial Intelligence Lab of the Universidad de Buenos Aires with Diego Fernández-Eslesac. We also work very closely with the Hospital Privado de la Comunidad, particularly with María Emilia Clement and Pablo Ioli. We work very, very closely with people from Universidad de la República in Uruguay, specifically from the CISEA group, Verónica Nin, Alejandra Carboni, Juan Valle Lisboa. I myself am from the Neuroscience Lab and CONICET in Argentina, in Universidad Torcuato Di Tella, and we, with my group, we also do all this with Melina Vladislauskas, Gabriel Paz, and all the other parts of the team. And this is all our funding. Thanks a lot for listening. You can find us in Facebook and Instagram. Just search for Mate Marote, and you can also find us in matemarote.org.r. I remind you, we are open for collaborations, and Mate Marote is free. So thanks for listening.
Okay, thank you very much, Andrea, for this talk. Very interesting. There's a lot of things that remain out of the talk of each one of you, of course. And there is a question in the chat, but I think it's a more general one that we can try to answer later in the general discussion. And I have a couple of questions. I don't know if it is too specific or too general, but which is the basis for the games that you choose for the Matemarote in this case? And I suppose this will be different when you want to apply to children and adult people.
Thanks for the question. It's a really important one. They are not different games, but different levels. The games are pretty much the same because executive functions are pretty much the same for all of us, except that we are in different levels of development. So, for instance, if you are playing with a specific inhibitory control training game, if you're an adult, your reaction times would be faster, and probably you would have better responses in general, better performance. So the levels of difficulty, the difficulty of the game should be targeted by your performance. But the games itself are pretty much the same that we are using for adults and for children. And the games have a specific goals. So for each of the executive functions, of course, the executive functions work all together, and we just try to build some games that use more of one of the executive functions in expense of the others. But of course, you are using all of the executive functions and also the attentional networks. So, for instance, you have games that to train planning. So, a planning game should have a specific. So would have an objective, and you should have to plan ahead in order to win the game, to get that objective. We also have inhibitory control games. So you have to. So the games make you want to do one thing, but you actually have to inhibit that behavior and in order to do another thing. So we also have working memory games. For instance, the classic, we don't have it yet, but the classical memory that we all have at home, that we all have played. So you have to maintain some characteristics or some locations for brief period of time, and operate with that. The important thing is that they are all the games are games. Actually, all the games are pretty much funny, and children and also adults enjoy to play with them. And they also adapt the difficulty level to each individual. So it's not as much as the age, as you mentioned, but as the cognitive baseline that each person has. So one person, so we are now actually trying to be working with machine learning algorithms in order to build personalized training protocols. And also always through games. We also test. All the pre-tests and the post-tests are also game-based. So
excellent. Excellent. Thank you very much, Andrea. We have many other questions that we can leave them for at the end of the symposium. So thank you at the moment. And we can go to the third speaker, Juan Carlos Valle Lisboa, with this talk, Cognitive Science is the Bridge Between Neuroscience and Education. Thank you.
Hi, my name is Juan Carlos Valle Lisboa, and I work at the Universidad de la República in Montevideo, Uruguay. I want to thank the organizer for inviting me to this meeting, and I'm going to talk about why cognitive science is the bridge between neuroscience and education. So the plan of my talk is first to refer to the ideas of a bridge too far, and then I'm going to move on to explain the connections between neuroscience and cognitive science and education with the example of reading acquisition. And I'm going to first go to a behavioral part and then to a neuroscientific part. And I'm going to finish my talk by connecting the three fields: cognitive science, neuroscience, and education. The idea of a bridge too far comes from this paper by John Brewer in 1997. And the argument by John was that although in the future there could be applications from neuroscience to education, at that time that was not the case. That was not the case. So that there were interesting approaches, but none of them had impact on education. After this landmark paper, several of the most important cognitive neuroscientists attempted to develop these applications. And we had in our region a reflection of that, because we organized together with Andrea and many others, several schools, summer schools, gathering the most important cognitive neuroscientists and to show what they and discuss what they've been doing about education. And in these schools, we organized one in Uruguay. We invited John Brewer, who was the president of the James McDonald Foundation that supported the schools. And we discussed this issue, and the idea was to construct the bridge. But nevertheless, there are still, within the psychological community, several skeptical researchers about this plan. So why are they skeptical is important to consider, and I'm going to move to our own research on reading acquisition. And in that field and many others related to education, it is true that the most relevant data, theories, and the methods about what it takes to learn to read come from behavioral-based studies. The idea is not that behavior per se is the only important thing, but the idea is that there are many information processing skills that need to be mobilized and combined to obtain a decent, an adult level of reading. And these are related to, as this classical paper from the 70s and 80s say, from phonological awareness, rapid automatized naming, which is related to lexical access. This lexical knowledge interacts with reading in a reciprocal causation loop, and that learning to read entails most, in the first part, spelling words. So there are a set of classical predictors of reading that are purely behavioral and that are based on theories that are purely described at the cognitive level without any mention of neuroscience, although some of the theories, especially the connectionist ones, were inspired by neuroscience. So we try to apply this paradigm to our own research. So we set to assess classical skills, those classical skills I was mentioning, and use them to predict which children will have difficulties learning to read, starting from kindergarten through grade 1 and 2. And we took advantage of the Plan Ceibal, which is an infrastructure in Uruguay that gives every child a laptop or a computer and gives access to internet to schools and squares and public spaces. And then we created a set of self-explanatory tasks to put into the tablets and do a longitudinal study with that. So what we did concretely was we started with a huge, for the standards of this type of study, a huge sample of more than 600 children in kindergarten, and we followed them until grade 2 in public schools in Montevideo. And we put into the tablets a set of games and applications that we can use to measure phonological awareness, letter knowledge, memory, etc. The important thing is that these things can be done, these applications of measurements can be done in a school setting where every child has his or her own headphones, and they can be playing this game in parallel in a classroom. And this allows us to measure a lot of children in many, many different things. The things I'm going to talk about are these ones, but we measure many other things. This is letter knowledge, rapid automatized naming, phonological awareness, and verbal short-term memory. And for reading, we measure 3 things, and also general cognitive and demographic variables. So what we did with this study was, after the first grade, I'm going to just talk about first grade, not second grade, we went to measure a composite score of reading related to decoding, fluency, and comprehension. We made the average score they get in these skills is the composite score. And what we see is that in first grade and at the end of first grade, there's a bimodal distribution with most of the children around the first peak, reading more or less around the expected level, but there are a sizable proportion of them that are indeed much below average. So we think that these are the lagging children, or children that are not reading well, or poor readers. And so we wanted to see if we could predict from the skills we measured at kindergarten the level of reading they will reach in first grade. So what we did is use this in particular short-term memory, phonological awareness, letter knowledge, and socioeconomic status. We input that into a model, and we try to predict reading scores or the reading category in this particular version. And we did a cross-validation study to avoid overfitting. And with this manner, we can reach 90% sensitivity and 76% specificity. But this has no neuroscience in it. So you can go a lot, you can move and advance a lot in understanding reading without any recourse or any mention of neuroscience. And this is the basis for the skeptical arguments. But there are theories that really connect reading with neuroscience, and let me go into one of them. So there's this idea that the brain rhythms that you all know about, delta, theta, and gamma, to name some of them, are related to rhythms that come in the sound and the speech signal as a sound signal. The sound signal as a speech is a continuous signal. You don't have evidence, you don't have a visual evidence, so you cannot see where words start or finish, or where phonemes start or finish. So the task that the brain needs to do when processing speech is to parse this and to segment the signal and parse it in the components and build and access with this the words. So the idea in this theory is that the process of the signal, which is related with the intonation of the sentence and phrases, is slow and around 1-2 hertz typical frequency. Then you have the frequency of syllables, and then you have the frequency of phonemes, the oscillations related to phonemes. And that this, for instance, delta can be entrained by prosody, theta can be entrained by syllables, and gamma can be entrained by phonemes. And the idea is that these rhythms in the brain are nested, so you can get prosody from delta by entrainment, and then this gives the boundary inside which the theta will find the syllables, and inside the syllables, gamma will find the phonemes. So the idea is that this is somewhat distorted in people trying to learn to read. So the phonological representations they get when they just are learning to speak, to process speech, gives them this set of phonological representations that are distorted. So they have a hard time learning to match this with the letters in print. The idea is that we should be able to see if there's differences in entrainment in the brain rhythms in children that learn well or children that learn slowly. The problem with this is that EEG is very expensive and we only can do this for a few students. We had a sample from this general study of 37, but then we had this other idea that maybe this entrainment also participates in the in other tasks, motor tasks, unrelated to language, that can be also and simply measured. So what we did is put children to listen to a simple beat and try to synchronize to the rhythm. They try to do so and they get better when they get older, but we can measure, using circular statistics, a form of non-linguistic rhythm sensitivity. And we can see if this non-linguistic rhythm sensitivity, we don't use the 4 Hz tapping rhythm, if we can see if this sensitivity is predictive in our models of reading. And indeed it is. If you don't put in the model the phonological processing steps or skills, you have quite a strong impact of tapping to a rhythm in reading. And also, if you also include verbal short-term memory and phonological awareness, you still see a contribution, but lower and less significant than in the previous one. So we believe that this shows that this non-linguistic rhythmic sensitivity is indeed related, and the relation is in part mediated by phonological awareness. We wanted to see what happens in the brain with this, and what we did was submit children to modulated white noise when they were just passively watching a cartoon. And what we see is that those that turn out to be good readers in first grade have a higher entrainment, a better entrainment at delta than non-readers. And this is mostly, the entrainment is seen in 4 electrodes after correcting for multiple comparisons, but the difference between the two groups is only seen in the frontal electrodes, and we're still trying to understand what's going on, and maybe working memory or attention processes top-down in a top-down fashion. They interfere with or interact with the processing of sound. We don't see that at theta. We believe this is mostly a problem with the syllables, which is with the prosody, which is related to other theories of how speech processing impacts learning to read. I don't have time to go into this. So the idea here is that we could use not only those classical predictors to assess reading, we can also use these other non-classical predictors, and these non-classical predictors can also be used in trying to intervene. We are doing a project I don't have time to go into, which is to use this French game developed to teach learning to read to children. We used it in a pilot study at the end of first grade. We didn't find any evidence of impact, but we wanted to move to the first month of first grade, of the last month of kindergarten, and from the evidence we know, they have an impact in that. And this is something we are doing at this moment. What I want to say at the end is that any attempt to use neuroscience to impact education should go through behavioral studies, and that the behavioral studies are based on theoretical interpretations that are based on computational theories that not necessarily talk about neuroscience, but neuroscience can have an impact at the level of the implementation. There's no obvious connection between tapping to a rhythm and reading if you don't go through the neuroscientific theory. This is the group that carried the project. This mostly was the PhD from Camila Sugarramurdi. Soledad is mostly working with the Kalulu, and these are my collaborators and financial support. Thank you all for being here. I'm going to answer the questions.
So thank you very much, Juan Carlos, for your presentation. And I was curious about if I understood correctly, this NLR, non-linguistic rhythm sensitivity that you identified and used in order to predict reading difficulties. Is it correct? Yes.
I'm sorry, I didn't get your question.
The NLR, the non-linguistic rhythm sensitivity is a tool that you identified and used for to predict reading difficulties.
Yes, it's based on the mean. You do the mean circular training to the tapping. You measure, you transform the tapping the child is doing, comparing it with the rhythm you're passing through the headphones, and you convert that to circular statistics, and you calculate the mean value of that and see the distribution, and that's the score. So if you have a random tapping, you have like 0, and if you have a non-random tapping, although you might not be in the same time, but you can be out of phase. But if you are good, this is the parameter we are using. This comes from circular statistics, and we input that into the model. And the idea is that you're not really, this is not expected from classical behavioral studies, in the sense that there's no reason in theory that why tapping to a rhythm would be related to reading, unless you have a theory that connects the two worlds that goes through neuroscience. So I think that neuroscience is important, maybe important to devise and several interventions or testing strategies, but you need to have a theory that connects this to the world. I mean, it is very complicated to say, okay, this is the brain is doing this and that, but how you use that to connect to the world. And as you've seen, what Roberto talked about and what Andrea, which I have to say, I only included my collaborations in that project. We have a very big project with Andrea, many other things, and I apologize if I showed that I didn't. It's just that project. What they showed is several things that somewhat you talked about empathy. Andrea talked about executive functions. People are asking what are executive functions. There are highly developed constructs within the theories that come from cognitive neuroscience and cognition in particular. So I think that's my main point. Cognition is very important. You need to embrace the theories, but neuroscience can in the same way can give new theories. I mean, in all sciences you need new theories, and neuroscience changes the way you see the signals and information is processed. So this is my point.
Cool. All right, thank you very much, Juan Carlos. We can jump to the fourth speaker and take the discussion at the end of the symposium. And the next speaker is Norberto Garcia-Cairasco with his talk, Building Citizenship with the Network Science, Arts, Education and Society, ScienArtES. Okay, thank you.
Good morning. My name is Norberto Garcia-Cairasco. I'm full professor of physiology at the Physiology Department of the Ribeirão Preto School of Medicine at the University of São Paulo. I'd like to thank Dr. Grace Moraes from Federal University of Minas Gerais for the invitation to give this talk, Building Citizenship with the Network, Science, Arts, Education and Society. I'd like also to acknowledge the masters I have in my academic life, Dr. Covian, the founder of the Physiology Department, Dr. Sabatini, my advisor at the Master and PhD, and all my students all over the last 3 decades of my life. Obviously, having this combination of philosophical, academic, and scientific views is absolutely necessary for my contemporary view on neuroscience and education. Making possible the interaction between neuroscience and arts in a neuroscience laboratory is not exactly a simple task. However, I'd like to say that that neuroscience and art combination or of integration has been for a long time either a dispute or at the same time a project, a target for several epochs in human civilization. It is possible that if we see our world, our universe as a pluriverse, that means the influences of several cultures, particularly ancestral cultures, the pluriverse could be seen today as a possible or different possibles, or the so-called pluriverse. Then, if we accept the pluriverse concept, we need to accept that different ancestral cultures are explaining where we live, what is nature, what is the role of our species, basically, inside that complex context. What is disturbing in this moment is that the history we know about ourselves is based upon a Western way of telling us the story, based upon a Eurocentric science, etc. So in the book, The End of the Cognitive Empire, the coming of age of epistemology of the South, Boaventura de Souza Santos talks about the ecology of knowledges. Some of those knowledges associated to other cultures were deleted, basically. So we need to have a more sort of wider interpretation of our planet where we live. And obviously, probably the oldest recordings of these are those in the caves. So the man in the cave means where we have the symbols used by our ancestors in order to portray daily life. Obviously, in other cultures such as Mayas or Aztecas or Aborigines from Australia or Maoris in New Zealand, and obviously in the European age of the Renaissance through Da Vinci, or in the dissections made by Vesalius or of others, even Da Vinci, in order to try to discover which were the substrates of our consciousness, of our sensory and motor interpretation, of processing. Obviously, we are putting together science and the arts at the same time with the development of the technology, particularly the imaging in technology. We developed dissections or virtual dissections in order to compose which were the substrates of both also cognition, consciousness, and sensory motor integration. And basically the combination of the two things are necessary. The ancestral cultures, the old views of the world and of the life, like here in Vesalius, at the same time in the 3D MRI high resolution, which means there is a need of the combination of those views all over the decades and all over the centuries. In those cases, basically we're trying to explain what is our consciousness, our more sort of not concrete exactly phenomena of our brains, but at the same time creativity, for example. In projects such as the antidisciplinary project of MIT at the Media Lab, you have a combination of art, science, design, engineering, and at the same time you have the openness to have arts inside laboratories. You can look at creativity using MRI, scans, PET scan, SPECT, some others, connectomics. So we are basically trying to look through technology and science, what are the origins of art. At the same time, if you look at the patterns which are repeated in nature, such as roots of orchids, for example, of plants, they are certainly truly similar to the patterns of dendritic ramifications of neurons. Those neurons that can be seen as molecular and cellular phenomena. At the same time, they are the part of the behavioral phenomena or the sensory motor integration needed for our behavior. So the composition of this, or the composite view, is basically a sum and addition of all these things. We can look at over the time, like a century, to dogmas such as those in science that can be accepted or modified, even removed by new views and demonstrations. Cahalver's, for example, confocal images demonstrating neurogenesis in adult brains. Looking at nature, you have those patterns, these ramifications, and at the same time you can play with them in our ateliers, in our workshops, looking for modeling of those forms in nature. How to reach people in society through the knowledge they probably don't know, through Brain Awareness Week, for example, which is a campaign in order to remove myths, to remove beliefs associated to ignorance. Going to the public places such as a park, going to fundamental or medium schools or high schools. Obviously, we bring our knowledge to the society, inviting people such as artists to go into the university. In this case, for example, trying to portray what an artist with epilepsy paints as a faces in circle, and an artist such as Elaine, who is working with us at the Brain Awareness Week, is painting exactly the same phenomenon, but through the art and his interpretation of a seizure. Both ideas here made at the main campus or in the park, for example, are today covers the best epilepsy and behavior journal in the world. So we are getting information from urban artists coming to the academy, which is another way of putting these bridges between the society and the cognition or the knowledge we produce in our university. So we need to get more contact with them. In the times of pandemic, for example, I use my drawings in order to make concrete resilience. We go to the places such as a park here in the Brain Awareness Week, the so-called SOS, the Brain in the Park. You can see the curiosity and astonishment of this child looking at our brain. You can see, for example, in the concept of art without barriers, this painter, which is a painter which is tetraplegic, Daniela Caburu, painting with the mouth. This is an enormous demonstration of courage and resilience. We go to the schools. In this case, we play first some discussions with 20 teachers or professors, and later on over 4 workshops with 20 students, and in other middle school with 80 teachers and 120 students. We did some workshops and hands-on activities with them. And obviously we added these to the symposia we did in our universities, such as this. Look at the theme, looking for epilepsy cure along centuries and across cultures, from demos and blessings to brain complex networks and DNA editing. So we are talking about the beliefs, the supernatural explanations of health and sickness, and at the same time we incorporate the knowledge coming from science. And obviously we can do, as we did last year, a moment of reflection, a multi-ethnic view of the world and of behavior, with speakers being Black people, Indian people, and Latin American like myself, exchanging information of our views in the world. A world that has been suffering because of our bad interpretation of our role as a species. I mean, our arrogance tells us that you don't care about the planet. So global warming, misery, and violence will be obviously the end of our planet. So dry brain, dry mind, dry country, dry planet, obviously. Well, then how could we make the change? Well, through education. This is an example coming from a primary school with 12 years old students. This is their statement. Since the frontal lobe is our social filter, it allows us to make critical thinking. Therefore, it is clear why human voracity will destroy the rest of the brain. The metaphor here is the brain as a planet. So you can see that the frontal cortex is the voracious part getting into fire everything which is the rest of the brain. As a consequence, if we are destroying our own planet, our only home, our own species will be also at risk. What it will be for us in the future will be just looking at the Amazonia through a shopping mall, or eventually to painted artists of the forest in a devastated planetary scenario. This is well and sadly illustrated by Banksy. Our history will be deleted because of our voracious behavior and arrogance. Our cave art being deleted, for example. Obviously, in that moment, Edgar Morin, 7 complex lessons in education for the future will be a nice counseling What will rest is to get educated for a planetary view, as in the ancestral cultures, Pachamama and Ubuntu. In Uruguay, you have a demonstration of a graffiti art in South Africa, for example. We need to train PhD students, as has been said in Johns Hopkins by Gundula Bosch, to be thinkers, not just specialists, and putting together STEM with arts, which means science, technology, engineering, mathematics, and the arts, for an integrated view of our lives. We can be nice scientists, critical, integrative, making use of models, a computation of microscope, electrophysiology, etc. For having arts all together with the science will be an even more interesting and collaborative way of seeing our science. So cover arts, for example, a nice complementary views of the science we publish in objective tables, results, data, statistics, etc. So training our minds to make drawings, paintings, obviously to write poetry, to make music, etc., arts in general. I do this, for example, in a workshop in my house, or for example, playing with tools from the laboratory, but at the same time being part of these arts and making exhibits at the university or probably in public places such as the park, at the fundamental schools, making collaboration with people in our campus, for example, from the nursery school, from the philosophy school, and from the studies made with students in remote mode today in the pandemic, making logos of events such as the Brain Awareness Week here in honor, for example, of our women in neuroscience, singular and hyper-connected. That means making the exercise of reflections of who we are, where are the asymmetries, where we can sort of make more collaboration between people and scenario, and obviously having this connection between the old ancient cultures such as those in cannabis, ayahuasca, psilocybin. At the same time, we have the new technology in artificial intelligence, for example, all together with their specific values, because they are products of our brains, of our minds. I have the happiness of having a scenario at home also with my wife, the cats, the felines whom we live in a lab who is a happy lab in which we produce science, art, and education, which are the most sublime products of our brains. We can face properly with them the COVID pandemic, misery, and asymmetries, because the challenge is to build worldwide empathy and solidarity. Because of that, I'd like to thank you for your listening to me, and obviously to the grant agencies, to the institutions to help us to make this happen in our university, in our country, in our planet. We need to get together with empathy, with solidarity. Thank you very much for your attention.
All right. Thank you very much, Norberto, for this talk about so many different things and the relation of science, arts, and the policy and the building citizenships, let's say. So I was curious about the relation, between let's say the ancestral knowledge and what we are here for. Let's say a form of science, neuroscience. If you could give some other specific example, in particular, I don't know, I can think about drugs that are used for traditional rituals, and this kind of this part of neuroscience studies that I may know a little bit more.
Okay, well, thanks very much, Francesco, and everybody. I think probably the simplest way to illustrate that is the use today of medicinal cannabis, ayahuasca, psilocybin, are the molecules that are ancestrally have been used, obviously without the stamp of science, because there was no science. I think 30,000 years ago, there was no science at all. So the question is, if you don't sort of stamp something saying this is science, in particular Western science, Eurocentric science, that there is no connection between the knowledge, the so-called ancestral knowledge, and what is being recognized today with our objective way, rational way, etc. So I'm not saying that there is no need to replicate, there is no need to make experiments, to propose hypotheses, and to do the protocols in order to demonstrate. But if you look at the ancestral cultures, at that time there was no the science we have today. So having the composite of the two things, I think is basically a sort of more global planetary view of our world. We are destroying basically because of our Western way of making our armament construction based upon an economical target. So there's so many things being written, books, etc. on that. Is obviously some political things on them and some philosophical on the other way.
Yes, yes, I suppose there is huge literature about all this, and some you showed in your presentation.
Yeah.
All right, thank you very much. And I think we can now begin with the general discussion, or these last minutes up to 12:30. And yes. Okay. So first of all, thanks to each speaker for your kind participation and the video, and to be here, of course. And my idea was to ask two questions that are quite general, and so that each one of you can give his own, her own answer. And then we will go through the chat where we have a very, very specific question or very general question. So the first question is if you can give us, you already gave in some case, what do you think is the more relevant or peculiar example in your own research field of translation from science, neuroscience, to education, and in some cases bring this to classroom? Or so if you can give us a peculiar example, or what you think is the more representative. And then the second question, let's say, is that I think for the public that we have, the general public that we have in this session is interesting, is what about regional initiatives? So in the context of the FALAN network, I think this is quite important, and of course international initiatives. So if we can begin with the example, if you want, for each one of you. We may use the same order as the talk, if you agree. So Roberto, if you can give us a peculiar example of what you think is more representative or more relevant for you in your own field.
Well, that's a very interesting question, because it relates a lot with what Juan mentioned in his talk. That is the controversy between how to build the bridge. How far is the bridge, and what's the role of neuroscience? What's the role of cognitive scientists, etc. If you take the example of sleep, for example, it's not my field, my direct field of work, but it's well known that adolescents, for instance, have a phenomenon called jet lag, a social jet lag. They sleep less than what would be necessary for their well-being and their cognitive performance. And this has been the object of studies of chronobiologists, of physiologists, many different kinds of scientists. And they advocate that classes could be would have to be delayed 1 hour for adolescents. The start of classes, instead of being at 7 a.m., to be let's say 8 a.m. or 9 a.m. Okay, that's a result, that's a proposition that comes from data, from physiological data about sleep. But in order to implement it, we have to rely on different other levels of approach. We have to consult economists, sociologists, people from pedagogy, cognitive scientists. So we have to be able to address different heuristic levels from molecules to society in order to do the real translation. So what I think is that the bridge has to be built by taking into account all different levels of approach in science. And this kind of example, this, how to deal with social jet lag of adolescents, is a good example because it goes from physiology of sleep to implementation of social measures in the school systems of every country.
Thank you, Roberto. Andrea, what's your opinion about this?
I actually like to say something else about on top of what Roberto has said, specifically talking about adolescent sleep, for instance. All that we know is mainly from abroad, right? So mainly all the research is done in the US and in Europe, and we are really lacking good research, not good research, right? The research that we do is very good, but we do lack research, neuroscience, cognitive neuroscience, and educational neuroscience research in Latin America. And that's why I think it's really important, this symposium, and that's why I think it's really important the chair that Roberto mentioned, and for instance, we just to mention an example, just because Roberto mentioned that, we have a work in progress with Dr. Juliana Leone here in Argentina and other members of the team, and we have just published that our teenagers, that Argentinian teenagers, sleep way less than what is recommended, and much way less than what is published in Europe and in the US. So that's why I think it's really, really important to build these networks that you also asked, Francesco. And that's why I think it's really, really important to start building all these collaborations, because we really have to inspire in the research and in the publications that are done abroad, but we do have to include our own Latin American perspective in the science that we do. And I think that fortunately that's starting to be done, at least for the last decade, or maybe less, probably less. So.
Very good. So there are not many groups working on this, but some important ones there are in Argentina and Uruguay also, and for sure in Brazil. And I am not of this field, so I have no all the knowledge. But okay, Juan, what's your opinion? What can you add?
First, I want to say that indeed there's an Uruguayan group, Ana Silva, Bettina Tassino, Nacho Esteban, working with I think they shared with Julia and Diego Olomec and all these people working in this particular chronobiological studies are starting to develop and gaining momentum. And indeed, I think that the Uruguayans are the ones that have the most distorted sleep, but I don't know. You know, this is a rivalry we have with our brothers and sisters. Now, going to the first question, I think that, let me go first to the second question. I think that the things we are doing, this symposium, Roberto's effort to have the UNESCO chair, the schools here, there's a moment of building this part of the bridge here in Latin America. And I think that what Andrea said, that we are not weird in the sense that we're not Western educated, industrialized, I don't know what they are, the developed countries. So this is very important. We've developed our own science with the tools we have. And we have the variability in the population is higher than in other places of the world. So I think this is a very important contribution for humanity in a sense. And we have several tools and ideas, and I think that this is a very important moment to start doing this, or to spread this, because this has been doing, we have been doing this for more or less 10 years, as Andrea said. I want to say something about the first question. I think that what I try to show is that indeed, if you want to go to education, the most important part might be cognitive or psychological processes. For instance, there's a very important set of results related memory research to education, but these are mainly psychological results. But, and this is very important for me, I think that the science of memory, the science of learning to read, the science of learning in general, is not, you cannot separate it, the psychological part and the neuroscientific part anymore. I mean, people working in psychology are doing MRI, and people working in neuroscience are doing complex behavioral paradigms. So I think this is the real message. There's a science of learning, of how this translates to humans. It's not as much developed as, I say, I don't know, neurophysiology of the neuron, but it is a science nonetheless, and it has results and has something to say. And I think that teachers and educators need to be taught or need to learn this science, in a sense. I mean, the science of learning in educational training is mostly Piaget and Vygotsky, and that's all. And I'd say, I mean, the science of development, and I'd say that this needs to be changed. I mean, there are many things that happen after that, and I'm not, I mean, they were giants, Piaget and Vygotsky, but they are not the only ones, and they don't have all the answers, and many things that they propose are wrong, and we know that. So this needs to be taught at the teacher training schools. And this is a very important and difficult part. And I also think that it's not just training a list of things you need to know about neuroscience, and don't fall for the myths and whatever. You have to teach the science behind that. And we published a paper trying to show our efforts in that part. Just show the teachers the science and what it takes to produce the science, what it says, and what the limits of it that are. Because I think that Roberto said something, that the connection between this milieu of innovation that is similar to biomedicine. And I think we should maybe avoiding the pitfalls of this system of innovation in biomedicine. We need to do something similar in education, and the teachers are the medical doctors, and they should know all the science behind that, and they should work in that way. And they won't produce all the innovations they need, and they need neuroscience, they need psychology, they need computation, they need economy, many things that go into the profession, but they need to know the science of learning. And I think this is something that neuroscience has to be worried about.
Thank you, Juan. Norberto, can you add something more? Yes,
well,
for sure.
I think, in my view, thanks. Everything that has been said from the point of view of science, creating the bridges, like Roberto said, between education and neuroscience. You have the educators and the people that work with neuroscience. And not exactly, you have the hybrids that I think are the ones that we want, that understand the two fields and at the same time do the proposals in order to get solutions. The point I raised is in addition to that, because just in the book of Boaventura Sousa Santos, the end of the cognitive empire, is being said that we just deleted everything else besides the Western, Occidental, modern view of science. And that's a pity, because we are destroying our planet because we ignore exactly what is a planet. We have the arrogance to think that we are the best
in the pyramid, we are the human beings. This is an anthropocentric, anthropological, anthropomorphic view of the planet. So the question is, the planet will be destroyed with our help. So education needs to go that way. So Edgar Morin goes in his recommendations for education for the future in a conception that includes not only the humans, but everything else. And are we doing that with our students, our children, our society? With the negationists we are assisting, we're listening to daily, are having that perception of a planet, of the only home that usually in Cosmos or Carl Sagan used to say is our only home, our only planet, etc. We have that dimension. So I think that education needs to go that way. And when you go to the next, the connection between arts and science, arts and education, usually are the things that the children or a child used to do naturally, arts, whichever is arts. There's painting, drawing, musical things, even writing things, poetry, etc. So I think we have in here, in this symposium, I'm so happy to be here, a mix of things that need to be together. All the proposals are absolutely needed. But I think we negate these over decades where we just got into a specific way of making our view of the world based upon an economical perspective, the growing, the empires, every time, every century, and basically we destroyed everything that didn't match that proposal. But Europeans at some point went to either Africa or Asia or came to America to Christianize everybody who wasn't Christian. But that is a story that began 2,000 years ago. And the other stories, which I was born in Colombia, I live in Brazil, I have 2 citizenships. I wasn't born in Japan. So what is the problem with that? Because I don't understand things that the people in Japan understand, looking at the world, the ways Japanese or Oriental people see the world. There is a contradiction that, no, not at all. We discovered that the Earth was round or a globe, etc., where we start beginning to conquer things. And after that, we just remade the conception with the conception of the winners. The history is being told by the winners. That is an old statement. You start, good at Ailton Krenak. Ailton Krenak is a writer, is an Indian from the Krenak culture here in Brazil. Go to the Yanomami, Davi Kopenawa. It's a beautiful story. There's a book on him, from him recently, talking about the Yanomami, one of the oldest cultures in Amazon. You need to delete that because this is not based on science. So basically, that's the contradiction I think we need to solve with our contemporary recent tools, but the incorporation of the old ancestors telling us about usually knowledge and particularly wisdom. Obviously, with the rules of science, you want to put science on it, but not exactly the science being made only by the Western conquerors. That's, I think, the point. This is something to be discussed in several other places. My video and the text I wrote and sent to Francesco in Spanish and Portuguese in English with 16 references. So you can take them and we can start discussing our Latin American network. I think we'll be adding things to the future.
Yes, thank you, Norberto. Yes, we will put the material hopefully in the FALAN platform that Norberto sent us. And thank you very much. So I would like to we are approaching the end of our session. And I know that many of you have not only work in her own region, but country, but also at regional level. So in the context, of course, of the FALAN network, I would like to know your experience, if you can tell us something about regional collaboration. And of course, we heard also about international collaboration, but just start with the regional one, please. No? Yes. I don't know, Roberto, Juan Carlos, Andrea, and Norberto.
Yes.
What about the regional initiative?
We do have, we mentioned something. I think there are plenty of initiatives, fortunately. And for instance, with Juan, we met, I don't know how many years ago, Juan, 7, 8, in a Latin American School for Education, Cognitive and Neurosciences, and which started some years ago, and with the goal of fostering a new generation of people that would actually build that bridge, that could actually speak both languages, the neuroscience language and the educational language. And actually that's really, really difficult. And I, for instance, I find very interesting what Norberto said, because he didn't mention that explicitly, but actually he was also talking about the bridge, and that bridge that is too far or is not too far, it depends. But actually we usually don't feel it, and we usually don't treat that bridge as a bridge, right? Because a bridge just connects two equally leveled fields, and usually neuroscience is considered like highest in a highest level as education, and we usually forget that, for instance, we know a lot. We don't know anything about how the brain works, but we know a lot. But educators also know way too much than us, for instance, about development. They for decades sometimes they see the brains evolving and we don't have that in the lab, nor even in our labs or in our settings, in our natural settings. So so I wanted to mention that because I think it's a very beautiful parallelism with what Norberto said and the bridge. And going back to the collaborations, few collaborations are now. Roberto mentioned something in his talk, and he might want to say something else now about the UNESCO chair. The science of learning is growing globally, fortunately, and is also growing locally. And a proof of that is that we have a symposium in the FALAN networking, right? And that's so the neuroscience field is actually starting to think that the science of learning is an application of neuroscience. It's a real world application. So I think the four speakers in here are very open for collaborations. We are already in contact in the networking of FALAN. So I think that I speak for all of us. Please don't hesitate to contact us, so we can work together.
Thank you, Andrea. So it's Norberto, would you like to say something about the regional collaboration? Or
I think it's the same line that Andrea Paula said. Obviously, yes, I didn't say bridge, but I'm trying to do a huge bridge between different things. So that's why I created that network. That initially was a neuroscience, contemporary neuroscience, whatever. So I said, okay, you need to have education in there, you need to have impact on society, etc. So there's a lot of different points to be bridged or connected. So I have collaborations with Colombia. Obviously, I was born in there, so I receive the students in my laboratory. We have some projects in CNPq Brazil that I think are not working now. It's the so-called ProSul. I received people from Colombia and from Argentina years ago. At the same time, I'm working with epilepsy and neuropsychiatric comorbidities. This is our line project in our laboratory. Is basically an interaction between basic science and translational things, it's clinical epilepsy and psychiatry. And every 5th year, we celebrate anniversaries of our laboratory. Is that since 1998. So every 5th year, we invite people from the whole world are working in that scenario, epilepsy and neuropsychiatry. At least 10 people of those speakers are from Latin America. And usually they don't pay the tickets, for example, they stay with us for the whole period of the conference, paid by our sources, FAPESP and CNPq. We hope we'll have CNPq, Robert, for the next years and decades. We are basically destroying. We're not, our government, sir, that's a complicated thing. So at the same time, we have these collaborations with visiting missions between people. They stopped because of the pandemic, but usually I go to Colombia, I go to several places. I went already to Chile, Uruguay, Argentina, etc. And we're open for those visits and collaborations and missions. Obviously, as Andrea said, I'm speaking to be part of that openness through FALAN. Obviously, in the truly academic interactions, science in our fields, but at the same time in that nexus or connection between science, arts, and education. Yeah. I think we need to work together to get stronger, to ask for money, even international, like NIH, Fogarty, European Science Foundation, etc. We need to get less shy, to ask people. But you have billions to be used going to the moon or going to get some rounds, Musk and Bezos, etc. Why don't deposit some in our FALAN's account, for example? I can make the suggestion for that.
Oh, we can apply for space funding for education.
Exactly. Yeah.
Exactly. So before closing, and I don't know, Roberto and Juan Carlos, if you want to add something about this initiative at regional level and UNESCO, for instance, or
I can add something. We have this project still not. We don't have an approval, a formal approval by UNESCO, but hopefully we'll be able to build this UNESCO Chair of Science for Education. It's a start for a network of more multinational efforts. But I hope that perhaps we can kind of build a Latin American network of science for education. We have this Brazilian network, which now has over 150 researchers of different disciplines, from economy to molecular biology, whose work somehow may apply or may be applicable to education. And why not having a Latin American network of science for education? We could be more, even stronger than we are right now. And maybe we could think about that sometime. Maybe in a future FALAN meeting, we could have a special symposium on that, and that could be the start of this Latin American effort.
I don't have much to add to that. I think that this is a good idea. I think that there are many things going on, and we should unite them behind some powerful network. And I think that this might be, given the diversity of our continent, might be also an important thing for the world. I think that we should try to create this network for Latin America, but also thinking about humanity in a broad sense. We have a lot of variety and many different things that Norberto was talking about, not just people living in nice cities in European countries. So we can show that and give that to the world. And I think we should build some kind of, following the lead of Roberto and the things we've been doing and many other initiatives around, we can create something, and hopefully in the next FALAN we have news and apply for money and do something really productive and powerful.
All right, excellent plan. I think in order to close this symposium, I hope and I have the sensation that this symposium has also contributed to the growing of the bridge between neuroscience and education, let's say. So I would like to thank all of you for your kind participation and proactive participation. and I hope we will see you again very soon in another initiative. And of course, I suppose FALAN, IBRO, and other organizations can help and support a regional initiative for a network of neuroscience and education. So thank you all, and to the participants and the public in general.
Thank you.
We can close here.
Thank you very
Thank
much.
you. Un abraço. Ciao.
Hi, everyone. Welcome to our last session before the closing. And our session is named Supporting FALAN. The organizing committee thought that we should have a time to do some thinking about how to make FALAN stronger and more active. Of course, as individuals, we can log into the platform and make use of it, like get to knowing people from Latin America and improving our connections. And apart from individuals, we can also learn about new initiatives and ways to think about funding. So we're going to start our session listening to Professor Daniel Olazabal. Thank you very much for accepting being here with us. Daniel is an associate professor at the Department of Physiology. So like me, I'm from physiology as well, at the Universidad de la República de Uruguay. And he is the actual president of the Let Brain Initiative, and he's going to present to us a little bit about this initiative. Thank you and enjoy our session.
Hello everyone. This video will introduce the Let Brain Initiative, an effort from several Latin American countries to better understand how the brain works. Over the last years, several countries have initiated government programs and brain initiatives with the aim of improving our understanding of the functioning of the brain and its mysteries. In this way, the US Brain Initiative was created in 2013, and the Brain Initiative of the European Economic Community that was given the name of Human Brain Project. These two initiatives were quickly joined by the Japanese Brain Initiative, the Brain Initiative from South Korea, and later the Canadian, the Australian, and the Chinese initiative. All of them with a very important investment in brain science. In 2018, the International Brain Initiative was also created, which is a conglomerate of brain initiatives whose vision is to catalyze and advance ethical neuroscience through international collaboration and knowledge sharing, uniting diverse ambitions and disseminating discoveries for the benefit of humanity. The main goal of all these initiatives is revolutionizing our understanding of the human brain, treat, prevent, and cure disorders of the brain. Some other goals also include, for example, to answer this question: how does the brain learn, remember, and adapt? To understand the principles of higher brain functions, brain development, and the dynamic changes from healthy to pathological brain. To establish multidisciplinary projects, and in this way to develop the next generation of tools needed to explore how dynamic pattern of neural activity in the brain controls thoughts, feelings, and movements. Also, one of the goals is to apply knowledge of the brain to improve education, societal and cultural well-being across the lifespan. Some other goals are strengthen studies of non-human primate brains that will directly link to better understanding of the human brain, to elucidate the neural network involved in such brain disorders as dementia and depression, to develop personalized treatment for mental and neurological disorders, to promote close cooperation between basic and clinical research related to the brain, and to stimulate the interaction between scientific institutes, academia, and industry. Finally, some other goals are to create a fourth industrial revolution driven by brain science, establishing an advanced neurotechnology industry sector, to apply advanced computer technology to neuroscience and medical research, to have access to shared brain data, the replication of brain architecture and activity on computers, that is called brain simulation, and to catalyze technology development and improved brain-inspired artificial intelligence. It is in this stimulating and rich context for those of us who work in neuroscience that a group of researchers from different countries launched in 2019 the idea of working to create a Latin American brain initiative. Our first meeting took place in Montevideo, where we signed a declaration of intent, which can be found at the following link, where several representatives of Latin American countries agreed to work to create the Latin American Brain Initiative. The following year, at the annual meeting of the Sociedad Argentina de Investigaciones en Neurosciences, we held a symposium that resulted in the signing of the founding document of the LAT brain. This founding document has been at the moment supported by 12 countries listed in the slide. This foundational document also established the mission and the vision of the LAT brain, being the mission to promote and advance brain research through collaboration and knowledge exchange, combining the different approaches and visions in Latin America to expand the scope of our research and thus produce and disseminate new knowledge and scientific application for the benefit of our countries and the world. The vision is to achieve a deep and comprehensive knowledge of brain that leads us to improve the quality of life of people in the world, in the different areas of individual and social life. The LAT brain will achieve its goal by identifying common priorities to the different countries and facilitating the exchange of ideas, recognizing opportunities and challenges in our region, building networks and catalyzing collaboration that involves its members, scientists from academia and industry, educators, philanthropists, communicators, government, civil society, and stakeholders from other areas of science, and stimulating the creation of national programs and projects in brain science. This foundational document also established a strategic planning steering committee. This committee will be formed with a representative and to alternate from each of the participating Latin American countries, and each country will resolve the coordination of the election of these representatives, which should include the societies related to brain science, local brain initiatives, or if they don't have them, the centers or institutions of reference in brain science in each country. This slide shows the current representative of the 12 countries that support the LAT brain at this moment. This foundational document also established a collective of participating nations. This collective will include organizations that participate in the LAT brain and support it, such as research centers, universities, and research laboratories, among others. Other organizations of recognized scientific activity in the area of brain sciences and civil associations or organizations that have adhered to the Declaration of Montevideo will also be part of the initiative. The document, finally, establishes a collective of companies and funding agencies. This collective will include those agents interested in collaborating and financing the initiative. All companies and funding agencies will be invited to participate in the annual meeting of the LAT-BRAIN. We are currently carrying out several activities. First, we are promoting the LAT-BRAIN. We will soon make a campaign requesting to join the LAT-BRAIN. We later will create working groups to develop our strategic plan. And finally, we will contact authorities and financial agents requesting to support the LAT-BRAIN. Finally, we want to invite you to join us in this great challenge. Thanks.
Okay, so thank you very much, Daniel, for the presentation. Let me just silence my YouTube so we do not get confused. We have some comments from the audience congratulating this initiative. I would like to ask you, in each way you think that FALAN can help to promote LAT-BRAIN, and how we can work together to make neuroscience and brain science stronger in our countries.
Hello, everyone. Thank you, Grace. Well, your question is very important. I have to say that FALAN was actually founder of the LAT-BRAIN. Elaine was in the first meetings. The president of FALAN was in the initial meeting that took place in Montevideo. And she has been part of representing FALAN of all the development of the LAT-BRAIN. I think FALAN will be one of the closest partners and collaborators and actually part of the LAT-BRAIN, as we hope will also join some consortiums, some other networks related to brain science or interested in collaborating with the development of brain science and knowledge about the brain in Latin America and the world. And I'm pretty sure, because many of the founders of LAT-BRAIN were members of scientific societies for neuroscience in the different countries of Latin America, that we will work from inside the organization. Actually, FALAN is part of the LAT-BRAIN, and it will be a great opportunity to join the societies for neuroscience of Latin America with other societies related to brain science, clinical societies, consortiums working in different issues related to brain science, other disciplines, because one of the goals of most initiatives is to have multidisciplinary collaborations and projects. So I'm sure that FALAN will be the main contributor of LAT-BRAIN, probably.
Yes, I'm sure this is a very nice and important initiative. Thank you very much, Daniel. And now, to keep discussing how we can support FALAN, I invite Professor Fernando Stefani. He is the Deputy Director of the Centro de Investigaciones em Bionanociências in Argentina. And Fernando has a very interdisciplinary career. So I could see, like, in his curriculum, he's from physics, but also chemistry, So he has projects in very related with technology. So I'm sure we're gonna love to listen to him talking about the Latin America pitfalls for funding. Thank you, Fernando, to be here with us.
Hello, my name is Fernando Stefani. I would like to thank the organizers, first of all, to inviting me today to the FALAN networking online meeting to talk to you about scientific policy. In particular, I will be talking about the scientific policy in Argentina, but I think that most of the concepts I will discuss today are valid to many other Latin American countries. So I would really love to hear your comments after my talk. Let us start. I will share with you some data. Here you have my contact information as well as in the program to send me comments and ideas after my talk. So first thing I would like to show you is this world map of global investment in R&D. What you see here are the countries of the world colored according to their investment in R&D, in research and development, expressed as percentage of their GDP, their gross domestic product. So that's basically showing what fraction of the economy invests each country in research and development. The first thing we can notice is that these numbers are small. They range from 0 to 4%. And the second thing we notice is that the countries that we know as developed countries are the ones that are investing more, like USA, European countries, Japan, Australia. And Latin America is a region that invests moderate to low values. Okay. The second thing we can do with this data is to see if there is this correlation that we have just seen by eye is expressed more emphatically. And we can see that, for example, by plotting this data. Now we have here plots of GDP per capita, so that's the average wealth of a country, as a function of their investment in R&D. And you see two plots. On the left, here we have data for over 60 countries in the period of 96 to 2000. And on the right, the same plot for the same countries for the period of 2014 to 2018. That's more or less today. And we see now that the correlation that we saw in the world map is clearly shown here in these correlation maps. So the correlation is clear and is sustained over time. We can take the same data. So these are two snapshots, but we can take the same data and try to see an evolution of these countries. That's shown here. We have the same plane of GDP per capita versus investment in R&D, but now the countries are shown as arrows. The origin of each arrow is where that country was in the period of 96 to 2000, and the tip of the arrow is where that country landed today. And also the countries are colored. So colored in red are countries that increased their GDP per capita more than $30,000 US. In orange, countries that increased their GDP per capita between 15 and 30,000. In green, between 8 and 15. Light blue, between 3 and 8. And in purple, the poor countries that increased their GDP per capita less than 3,000 US dollars. So what we see now is that there is an order here. Even though we have a universe of countries, very diverse, in very different positions in the globe, geographical positions, geopolitical positions, very different levels of natural resources, very different internal organizations, and very different policies of distribution of wealth. That doesn't matter because we are talking about average wealth. We see that there is an order. So all the red and orange countries are over here. then we get the green countries, then the light blue, and then here all the purple are grouped together. Argentina and most Latin American countries are here in this region, investing small numbers in R&D and also increasing their GDP per capita very little. On this plot invites to analyze individual cases like the case of Norway or Australia, how they managed with modest investments of 1.5% of their GDP and managed to obtain very large increases in their GDP per capita. Well, now the key in these two cases lies in the high level of natural resources per inhabitant, but there are other cases like Switzerland, etc. Japan, for example, is a pathological case that suffered deflation in this period for excess of offer, and that's why they couldn't increase their GDP per capita, etc. But nevertheless, the correlation is clear. Richer countries in average are the ones investing more in R&D, and also the countries that invested more in R&D are the ones that increased their GDP per capita in higher amounts. Okay, so this relationship is clear. But now the question still arises, what's the cause? What's the direction of this arrow? So higher investment in R&D leads to a higher wealth, or is it the other way around? In Argentina, it is often heard or thought that this arrow goes in this direction. We often hear sentences, statements like this, like developed countries invest more in R&D because they have the money, they have the resources, because they already managed to satisfy their priorities, then they can invest in science and technology for tradition, for culture or fashion or whatever reason. Another way of expressing this thought is saying that in a country with certain level of poverty, like in Latin America, it cannot be priority to invest in science and technology. That's another way of saying the same. And that's a problem. There's a problem with this kind of thought because they are false. They are absolutely false, and they constitute a conceptual trap. We will see that thinking like this is a trap because it argues poverty not to invest in the solution of poverty. In fact, developed countries invest in research and development, in science and technology, because they are in a highly competitive race to transform scientific advances in economic successes. And there are numerous reports like the ones cited here that explain this. But there's one way to understand this that is very simple, and is a key concept and usually underestimated in our countries, a rather obvious concept, that a sustained economic growth requires a constant improvement of economic activities. That's very important because, at least in Argentina, it is often, the economic discussion is often around how to administer the available resources, how to administer the interest rate, the exchange rate, how to manage the activities that we are doing since over a century. But we are not thinking, we are not discussing how to improve our economic activities at a competitive rate. And if we don't do that, there's no chance to sustain an economic growth. And of course, the only way to obtain economic activities better, improved economic activities, is through new knowledge, new research and development. That's what enables the better economic activities. If you cannot see this so obviously, we can make an exercise and think in historical times to see that all economic activities, all of them, some faster, some slower, but all of them lose value in time. Think, for example, of the industry of ice. At the end of the 19th century, it was a business, it was actually a worldwide business to mine ice in the mountains and distribute it all over the globe in ships. So you could have a cold drink in India or the Caribbean islands with ice that was mined in Boston, for example. So that was an industry, worldwide industry, distributing ice. What happened? Did we stop consuming ice? Of course not. We consume more and more ice, but the mechanical refrigerators first appeared and then the electrical refrigerators, and mining ice became an obsolete activity. So, and you can think of other examples like the photographic film. Kodak was enjoying of practically of a monopoly during the 20th century of producing and selling photographic film. We didn't stop taking pictures. In fact, we take more and more pictures, but selling photographic film became obsolete. And that happens with all activities. So you can think of any example, and sooner or later, first they lose value because the economic reproduces as other people can do it. There is a larger offer, so the relative price goes down, and eventually the activity becomes obsolete because a new technology displaces that activity. So this is expressed, what I just told you is very simple, and then that also explains why developed countries invest in R&D. So imagine now you have a country or a region, and somehow you managed to put it at its maximum capacity. So a country basically involves a certain population, a certain industrial capacity already installed, and a certain level of natural resources. So imagine you put everybody to work doing the best they can do. Imagine you have all industrial installed capacity working at its maximum capacity, and you are also exploiting your natural resources in the best way. So you managed, you already reached your maximum productivity. Can you stay doing that for the rest of the time and aim to keep your wealth? Well, the answer is no, because the activities that you are doing are losing value in time constantly. So if you do not improve your economic activities, your wealth will go down in time. So that is the reason why the developed countries are investing in R&D, because they want to sustain their competitivity, they want to sustain their economic growth and their wealth, and the only way to do that is to develop new economic activities all the time. And to do that, they need new knowledge, new developments, new technology. Another way of seeing this is taking a look at the global product space. So that's, you can see it in this website maintained by the University of Harvard. It's very interesting. What you see here is a global product space. So each dot here is a product, and the lines between them are the links between products. So in the center of this map, you will see the more complex products. These are products that are depending on other products. So to produce a product here in the center, you will require other products that are simpler, that are in the periphery, okay? So let's take a look at USA. These are the products that USA exported a few years back in 2019. And you can see that USA more or less covers the complete product space. Here there is a bit of emptiness, and that's textiles. So USA doesn't seem to be doing too many textiles, but it's covering most of it. Let's take a look at Germany. Very similar, even more populated. Germany is a very diverse industrial country. But let's take a look at Spain. Spain cannot be considered an industrial power, but it's a highly diverse economy, as you can see here. And now let's take a look at Argentina. You see the contrast is quite clear. Argentina is not covering the center of the product space. It's rather diverse, so we have dots all over the periphery, but we are not producing complex products. And that's a problem, because the complex products are the ones that are more valuable, and also these are the ones that involve more work, more jobs, because they require a chain of production. The same holds for many Latin American countries. As you can see here, examples of Brazil, Chile, Peru. Mexico has a higher diversity, but still not complex enough to compete worldwide. And how to produce complex products? Well, the key is technology. Technology involves basically three parts that are hardware. Hardware are machines, infrastructure, software, that's codes, protocols, methods, and brainware. The brainware is the know-how. The hardware and the software are rather easy to transport all over the world. You can import machines, you can send software protocols over the mail, no problem. But the brainware, the know-how, that has to be learned. That's not, you cannot buy it. Increasing your know-how is hard. It's the hardest part because that involves learning, that involves sending people abroad to learn and coming back to implement and deploy the new knowledge or inviting experts and learning, etc. A few words about how technology impacts the economy. There are basically three main mechanisms, and each mechanism has a different degree of impact and a different time to impact. The first mechanism is the use of available technologies. That's quite simple. We couldn't be having this conference today if we were not using an updated operative system in my computer or new applications in my computer. All countries do this to some degree, and that's only a survival strategy. You cannot survive if you don't upgrade your technologies regularly, more or less regularly. But when you do that, there are always two options. You can buy the technology ready to use, or you can develop it locally. You always have these two options that have advantages and disadvantages. Buying the technology ready to use may be faster, may be cheaper, but it generates dependency in terms of future upgrades on repair, spare parts, etc. Developing the technology locally enables to learn in depth how that technology works and to adapt it to the local needs. Normally in countries like Argentina or Latin America, the technologies that we use were developed somewhere else in the globe. So it is important and often advantageous to adapt them to our markets, our needs. And if one does that, then the door is open for the next mechanism, that is innovation. Innovation consists of generating a product or a service that is new, that it uses a new technology or that it combines in a new way available technologies. But to do innovation, you need to understand the current technology. There's no way to do innovation if you do not understand current technology, because one can only work in science and in technology on the possible adjacents. We can only imagine things according to our current knowledge. So it's a learning curve. And then there are technological revolutions. These are events in the history of humanity that change completely the lives of millions of people or of the whole humanity. For example, you can think of antibiotics, you can think of monoclonal antibodies, you can think of the telecommunications, you can think of the microelectronics. These are technological revolutions. You can think of the laser, for example. In the past, the technological revolutions happened more or less by serendipity, by a positive feedback between technological innovation and scientific knowledge. Suddenly, a new technology arises that can change completely the lives of millions of people. But nowadays, developed countries are applying policies that produce technological revolutions practically in a planified way. So we know that, for example, the quantum computing and quantum information technology will happen. That will be a technological revolution. We do not know exactly when, but we know it's going to happen. So there is a plan nowadays to produce technological revolutions. That's done through the large initiatives, like, for example, it was at the time the space race, then it was the nanotechnology initiative, then the human genome project, the brain initiative now, and et cetera, et cetera. And that's important to understand because, especially in Argentina and in Latin America, I have the impression that we are not understanding the current great challenges that humanity is facing. There is a change here in the great challenges that we are facing. In the past, humanity was faced to great challenges imposed by nature: how to eat better, how to survive, how to improve our health, how to improve our transportation, our houses, etc. But nowadays, we are facing challenges that are produced by our own technological advance, like the climate change, the energy supply, the informatic and commercial globalization, the artificial intelligence, a growing and aging world population. There's a possible global resistance to antibiotics. There are pandemics. The quantum computation and quantum communication will generate a global revolution of all the e-commerce as soon as a quantum computer works more or less reliably. So all these are great challenges imposed by our own technological advance, and their solutions or the response to any of these challenges will come also from our knowledge and technology. So any new technology addressing these challenges will generate a new and highly and a rapidly growing economic market of global dimensions. So that's why it is important to understand this and start working in these directions. And in Argentina and in Latin America, we are not doing this with the magnitude that will allow us to be competitive in the near future. So let us take a look now on how we are doing. This is the investment of Argentina in R&D, expressed as a percentage of the GDP as a function of time. And we see that we were at the 0.4%, then we had some increase to reach 0.6%, then we went back down. Okay, these are ups and downs of our local policy. Let's put this curve in the context, in the Latin American context first. And this is what we get. Now we have Brazil up here. We have Mexico, Chile, Uruguay, more or less showing similar trends. Brazil is the only one that outstands with a higher level of investment. But now let's put this data now in the global context. And now I kept only here Brazil and Argentina to make this plot not too crowded. And as examples of developed countries, I put here Germany and USA, these two curves. And as examples of developing countries, I put South Korea, the purple line, China, and Malaysia, the green curve. So this plot already enabled us to make a classification of countries. Normally, we talk about developed and developing countries. That's the usual distinction. But there's no clear criterion to classify a country as developed or, more importantly, as developing country. So I would like to show you here that this kind of analysis allows us very easily to identify developed countries, developing countries, and stagnant countries like Argentina. The way to distinguish them is by means of their average increment rate of their investment in R&D. The total investment in R&D of the countries is not only different, but it also increases in time. And increases in time with a certain rate. Developed countries, here I have Germany and USA as examples, increase in time with an average rate of 0.03% of the GDP per year. Developing countries go faster than this because they are aiming to improve their economy, improve their economic activities at a rate faster than the one imposed by the developed countries. The developed countries are the ones that sell us the new technologies, the new products that we consume all over the world. If you want to be better than that, you need to improve your economy faster. That's as simple as that. There's a race. If you go slower than the developed countries, like Argentina or Brazil, then there's no way that you can pretend to be a developing country. In fact, you are accumulating technological retardation, and your technology becomes of less and less value in the global context. A curious thing is that the most important national initiative of China is called Made in China 2025. And if you see where the curve of China crosses the USA, is pretty much in 2025. So here you see the projections. These are linear regressions of the data, and you see that each country follows a linear regression pretty well. So these are policies. Look at China, look at USA, even Argentina, with its ups and downs, it has a policy of remaining low. Our average is 4 times lower than Germany and 2 times lower than the USA. So there's no way we can improve. There's no way that our products and services become better in time. So what to do? Well, first, if you want to revert this, you need a competitive increment investment rate. So you need to increment your total investment in R&D at competitive rates that can range from 0.06 to 0.12% of the GDP per year. That's what the nowadays world is imposing. You cannot choose this totally freely, but you have to choose it looking at the global context. I included here the example of Malaysia to see that you don't need to be a global power to do this kind of policies. You can do this at your own dimension, because what matters is the fraction of your economy that you are reconverting, not the absolute investment. So the next thing to know is how to invest, how to do this investment effective. And to do that, you need to understand the roles of each actor in the innovation process. So generating new economic activities of higher value requires new knowledge that is generated at universities and research centers, and you need enterprises, industries, the demandant of new technology that will apply the new technology in products and services commercialized at scale in your society. So these are the two actors, the research at the beginning, the companies at the end, and in between them, there is a gap. There is a gap, and it's a natural gap, because these two actors have very different roles. Researchers have the role of generating new knowledge. Companies, industries, even the public sector has the role of using this knowledge to generate new products and services that are better than the currently available. And doing this process takes time. So generating a new product or service at scale can take between 2 and 30 years. These are long times. And in this process, it is important to monitor the process with these three parameters you see here. The TRL is the technology readiness level. This measures if a new technology is useful. That's all. This was developed by the NASA in the 80s to manage their innovation process in the space race. They only wanted to know if a new technology was useful in the outer space. They didn't care how expensive it was, if it was possible to produce it in series or anything like that. They only wanted to know if it was useful. That's technology readiness level. Companies they wanted to use that, but they had to complement it with manufacturing readiness level. This index measures if that technology can be introduced in a product or service in a production environment. When you do that, you know how you're going to introduce that technology, and then you know how much it will cost. And with that information, then you can do a market analysis and check if that technology that is good, that it can be implemented in your production, can also be profitable. So companies invest in new technologies when these three parameters become positive or the risk is low. When they know the technology is good, they can use it and they can earn money with it. So this gap is natural. It's not an Argentine problem. It's not a Latin American problem. It's a natural gap. Our problem is that we don't understand this gap. And normally we blame either scientists or entrepreneurs. In Argentina, for example, it is often heard that the reason for this gap is that our scientists deal with very irrelevant topics. Another way of saying that is is blaming the companies, and they say, no, our companies are too miserable and they do not invest in technology. So these two statements or thoughts are wrong again, because these are assigning responsibilities to the wrong actor. A scientist has to do science, excellent science, that's the role. And a company should use technology when it's useful for them and they can earn money with it, not in other conditions. So this gap is closed by innovation. And innovation nowadays is not a simple task. It's actually a very complex and interdisciplinary task that involves not only knowledge about technology and science, but also about markets, prospective, fabrication, regulations, and even international lobby and geopolitics if we are talking about sensitive technologies like in defense or transportation or energy. So innovation is a very complex task, and developed countries do this, impulse this with specific intermediate institutions. Here you see some examples. Another important aspect to understand is that the research in all the world is financed with public funds. And innovation companies is mostly financed with private money. And innovation is usually in these institutions financed by public-private cooperation. And we can discuss in detail how this is done. Let me speed up a bit and show you some examples of these intermediate institutions. Probably the star institution of innovation is the Fraunhofer Gesellschaft of Germany. It was established in 1949, and now it has over 70 institutes, and they do innovation in practically all areas of industry. In Taiwan, there's the ITRI, the Industrial Technology Research Institute. It has been recently reorganized to work in 4 large areas: smart living, intelligentization, enabling technologies, quality health, and sustainable environment. Those in line with the great challenges that we are facing. Japan also has it. Let me spend some minutes on USA. USA, USA has an interesting story because they underestimated the value of manufacturing and its impact on innovation during the 70s and 80s, and now they are trying to revert that tendency. And in 2014, they established the National Network for Manufacturing Innovation, mainly inspired in the Fraunhofer Gesellschaft. And up to date, they have 16 institutes working on these topics, again, well in line with the future and current challenges of humanity. The same happened in the UK, very similarly, again, inspired in the Fraunhofer Gesellschaft. They launched the catapult centers, working on specific areas where they think their future competitiveness relies. You can see here cell and gene therapy, connected places, energy systems, medicines discovery, offshore renewable energy, etc. So what's the role of the state to make this work? Well, you see that the research is publicly funded everywhere. The industries need technology and innovation, but they cannot do it alone. They need support from these intermediate institutions, and they also need proper incentives to invest in R&D. So the state needs to collect money from the companies and invest it in research and intermediate institutions of innovation, specific and targeting specific sectors, and also has to use that money to deploy a broad menu of incentives for the private investment. If this is done properly, then the whole system grows and the companies start doing better products, better services. The state can get more money from them and complete the circle, making this grow until it reaches a kind of steady state. About the private investment incentives, there is a very interesting survey made by Deloitte about what are the incentive programs in different countries and which ones are effective and which ones are not. And this is a very interesting report because it is done from the viewpoint of the clients. So it's very clear why and which programs do work and which ones do not work and why. And finally, I'd like to show you this plot to see how this process develops in time. What you see here is the investment, the total investment in R&D of South Korea, that's the blue curve, from 1964 to 2004. You saw in previous plots that this has continued to grow, and now it's about 4 point something. But the interesting point is that at the beginning of this process, South Korea, the investment in R&D in South Korea was mainly all of it public. So sorry, this is the other way around. This is public and this is private. So it was 90% public. Over time, with a continuous policy, the private sector was taking more and more a higher and higher fraction. And that took over 30 years to get to 50-50. And then a rapid change occurred and a kind of steady state established where the public sector invests about 25% and the private sector about 75%. That's nowadays the steady state relationship in all developed countries. So Argentina is nowadays over here, but we are not moving. That's the key point. We are stuck here. And many other Latin American countries are around this area as well, where a science and technology system is maintained, but with a weak link to the real economy and the production. And that's the problem where this cannot grow and the budgets are cut and improved with no regularity and not sustained in time. So with this, I'd like to finish. And for those interested, you can see a small text that I wrote. You can download it from this website. And as I said before, please send me your comments about the situation in different Latin American countries. And now I will be happy to answer any questions you may have.
Thank you very much for your talk. And it was very inspiring in such a way that make us think that we are not isolated from the rest of the society. I mean, we have to think politically and bigger, and so very nice. We have some comments in the chat I would like to share with you both. So Deborah wrote, how can we convince the governments that science is an essential investment to improve economy? We've been trying to sensitize the Brazilian government to understand this basic concept without success. I'm sure you don't have an answer for that, but we'd like to hear what you think.
If I had it, I would be implementing it. But I think normally governments that lack the strategic vision of understanding this are impossible to convince. So because they are reacting to immediate demands, they are reacting to urgent problems, and they are not having that strategic vision that you need to plan your future. So I don't see a possibility of convincing politicians that have an horizon of 2 years or 4 years to the next election. That's very difficult. So what I think we can do is involve actors that have longer terms objectives, like companies or entrepreneur societies or labor unions. So these are actors of the society that remain. Governments change, but the labor unions are the same, and they will knock the door of any government to claim for the salaries or working conditions, right? So if they understand this, they could knock the door of any government to claim also to sustain a strategic program to develop local technologies, science, and better products, because that will mean for them better jobs and better salaries. So I would go more in that direction to do the efforts. But for that, we need to understand also our own role. I think now sometimes we are asked, so why should a government invest in science? And they ask that to a neuroscientist, for example, or to, and then if you don't promise that you will find a cure for Alzheimer next month, then you're useless, right? And then we start trying to say something that looks interesting, and that's wrong. That's wrong because that's not how it works. That's why I try to explain this, what the role of each actor, how long it takes, and try to explain this to the whole society, and also to actors that remain active through government changes. I think that's more important.
Yeah, we keep saying that here in Brazil, and I'm assuming in other countries as well, that we need to make the institutions stronger, because then they don't. If the person in front of the job changes, which will happen, the institution is strong and keep it going.
Yes, that's true. But with clear concepts. That's why I emphasize on these wrong concepts that are, around, like blaming scientists or companies or thinking that you cannot invest in this because you're a poor country. So these are concepts that constitute mental traps, conceptual traps. And so we need to clean that up. And yeah.
Okay, we have more comments I'd like to read. So from Claudia Garcia, I think COVID-19 pandemic has demonstrated the importance of science. However, governments from Latin America must invest and do better. This profile from Physiologia, UFSJ, no financial support, no hardware nor technology. Although simple and straightforward, money is necessary. Know-how doesn't do miracles, unfortunately. Claudia again complaining about the current Mexican president that believes in austerity. Good science requires funding. And then we have also a suggestion from Orlando Serrano Barreira. Through FALAN, we could exchange info on funding calls in the sort of map of technology available at each site. So the network could establish collaborations according to each one's capabilities. And then there's a lot of comments on thank you both and very important analysis and reflection.
Okay, thank you. So maybe we can comment on I also hear here in Argentina very often that COVID-19 taught us the value of science and technology. Okay, it's fine. But that's because we are just looking at this, but this, we could learn that just reading history. We don't need to experience a pandemics, right? So what we learned here, just because we are not paying attention to history, is that if you have a short demand, like it was in this case for vaccines or tests or other consumables related to a massive consumption, imposed by the pandemics, then you can diminish the risk of innovation and make new products that will be profitable. But this has been done since the Second World War in the developed countries. So after the Second World War, all the developed countries understood clearly the power of technology to power the economy. And since then, they have large initiatives and R&D programs to do this. And I mentioned some of them in my talk, like the space race, the nanotechnology, the Human Genome Project, the Brain Initiative, etc. So that's a learning. I mean, we can learn that from the pandemics, but we can also learn that just reading history, and it's there. It has to be understood. And concerning austerity, that's a usual excuse. And of course, austerity, who is going to be against stupid expenses? So nobody wants to spend money on useless things. But it's not about austerity, it's about strategic thinking. It's about where are you going to invest to be better tomorrow? And so that has nothing to do with austerity. That should never be confused.
Yes, yes, that's right. We could keep talking like hours and to come up with ideas and sharing experiences. That is something that really unites us all in Latin America. I'm not sure if I can ask now Danielle to come join us, and then we can. Can I?
I am here. I don't know if I'm online.
Okay. You're already here. So I'd like to thank you both. And let me see if we have more comments. Yes, we have a lot of comments. Great presentation. Thank you. And you want to say anything else, Danielle, about the LARC?
Well, just adding to the conversation, I think we also have to pay attention to keep working on the whole society and trying to understand. This is one of the goals of FALAN also, trying to make people understand the importance of science, because they can push government also to invest in science, I think, and we should keep trying, just working with common people, not only government.
Okay, they are calling my attention that I have to read the comments. So we have, I'm not sure about time, that's why I'm so confused, but we have here, where do you think is the misconnection between the national councils of science from each country and their respective government? I think it fits to Fernando, or both of you can comment on that. Have you understand?
What the question is, why there is a misconnection between the government and the Academy of Science? I didn't understand very well.
The national councils of science from each country and their respective government.
Well, in the pandemic in Uruguay, there was a very good connection between the scientific community and the government. It was an exception, actually, that now is trying to expand to other areas. So, but this is exceptional. It didn't happen too much in the past. But the pandemic was something that came urgently, and I think that is what pushed the government to get close to science. And we shouldn't wait to something urgent that happened to get, to get together to work to solve a problem. I think mostly the answer to the question is part of the talk of Fernando, what he has been talking already about the difficulties to understand certain things and the importance of science, and the narrow view that sometimes we have to get applications and solutions rapidly.
Yeah, I agree. Especially for us in neuroscience, yeah? Usually we think that we cannot solve like big problems like climate change. It seems so far away from us, but maybe we should stop and think that about a problem that we can unite, all of us can unite and help to solve, like being active actors, yeah, in this complexity. So I think now it's time for me to finish our session. And I'd like to thank again both of you, Fernando and Danielle, for sharing with us your thoughts. And thank you, the audience. And now I must finish. Yeah. Thank you very much.
Okay. Thank you, Grace. And thank you to FALAN for organizing this event.
Bye.
Thank you.
Bye-bye.
Okay. Buenas tardes. Boa tarde. For all of you, it's very sad to be here to end this meeting, but it is something really important for all of us. And it was very nice, this meeting. And we from FALAN have some things to show to you today, okay? First of all, look how many countries we have in this meeting, just 3 days meeting. You can see a lot of people from Colombia, Argentina, and Brazil. Also from Chile, Cuba, Mexico, Peru, Uruguay, and so on. And you can see the distribution around the world. So it was really, really a multi-country meeting. We are very happy with that. And you can have a look here also, no? Oh, sorry. How many non-member researchers? Sorry about that. Member researchers from some societies, from the societies. We have a lot of master students, non-member master students, sponsors, exhibitors, companies, members, postdoc, non-members, PhD students, a lot. I hope in a while they will become a FALAN or some neuroscience members. Please come with us, member PhD students and non-member postdoc. So that is the opportunity. You saw how good is science in all Latin America. So please come to FALAN, come to the Neuroscience Society of your country. We need all of you, okay? And you can see here again or not. Yes, 20% of investigators from Argentina, 7.8% from Brazil, Chile 3.1, Mexico 2.4, from the two societies 2.8, Uruguay 6.4, and Colegio Colombiano, congratulations, Zulma, 4.3%. So, and people from no one of these societies, a lot of them. Again, come to FALAN, come to the society in your country, okay? Going on, as our new code of conduct and the procedure of FALAN, you can see that we have equal numbers of males and females in the plenary symposiums as chairs of the symposium. And also, I believe it's discussing politics, no? So we have a goal, and this goal was completely fulfilled. Men and boys, girls and boys, all of them in equal number. Okay, so yesterday we can just comment with all of you that we were successful in approve the code of conduct. We proceed to change the authorities, and so very soon you will know about the new FALAN committee. We have people from Colombia, Argentina, Uruguay, Brazil, Colombia, Argentina, Uruguay, Brazil, and I do not remember. It's Brazil, Argentina, Chile, okay? So let's hope that they will organize the meeting next year and they will be very successful in their job, okay? The FALAN budget also was approved, and it's important to remind of you that we have the FALAN networking. We need to know each other, so please go to the platform, put your data and the papers and meetings, interest, and everything that it's good. Even you can put, like say in Brazil, you can teach the Chileans how to prepare caipirinha, and they can teach us how to prepare pisco sour. No, it's pisco from Chile, no? Or I don't know from where, Peru, no, I don't know. Chile, that's good. And just for you to know, the symposium and talks will be in the platform for at least 1 month. So if you want to see some of the symposiums or talks or discussions again, just go there. If we have a lot of demand, maybe we can live for longer, but at least for 1 month, okay? So just to remind you of the FALAN-IBRA-LAC.org networking, please go there and help bringing Latin America and Caribbean neuroscience together. Don't forget it. There was some very nice comments. One from Grace, no? This meeting is showing me that we are something good in Latin America, and we need to value our work more, instead of thinking that only what comes from USA and Europe is worth reading and citing. We also need to exchange more skills, training students with Cotutela, it's very good programs, act more politically in our countries to have more grants opportunities, maybe more public diffusion of the impact of neuroscience to the society. That's the only way we are going to get more money from government, I guess. I just would like to refer to the last talk that we had. He was talking to us, indicating to us that maybe the government is not the exact point that we need to go to got more money, more financial help, and so on. Maybe the laborers, the group of the laborers, because they are always there, and they are the ones that are unprotected. We can't forget, we Brazilians, that the only guy that was a president, he was from the Labor Party. He was the guy that created a lot of universities, a lot of positions for PhD students, and he put more money for education, okay? So I think it was very clever, this idea, and they are always with us. And there is another message from Christian, Chile. He's the president of Chilean Society. Grace is rising, important point. We should commit to fostering relationship among us. I hope we can maintain this wonderful momentum until the Belém meeting. Thank you very much, Grace and Christian, and all the other people that just send us very nice messages. That is the FALAN espíritu. So it's what I wrote. Me gustaría llamar la atención que espíritu, este entusiasmo está la unión, el objetivo de FALAN. Por eso hay que invertir en FALAN, invertir en la investigación en Latinoamérica, aplicar más para los grandes de LARC-IBRO. As part of the LARC-IBRO committee, we need to have more applications. So please just look at that, pay attention on it. I don't know if you can understand Portuguese, but a great writer in Brazil, I'm too nervous, so I won't remember the name of the guy. He said that Brazilians have a complex of vira-lata. Vira-lata is the dog that doesn't have any pedigree. I don't know how to say, to refer to the dogs of in Spanish or in Castellan. If you can help me, I would love. But you know, we always think we are the worst and the North Hemisphere are the best ones. We need to stop with that. And that is the message of what Grace and Christian told us. I don't remember the name of the guy. Okay, so to finish, because I don't want to take a lot of time from you, we need to talk a little bit. We would like to say thank you very much for the work of the organizing committee. Patricia Cassina from Uruguay, Vice President of FALAN, Paola Hager from FALAN, Treasurer from Chile, Maria Eugenia Pedreira and Pablo Lopez from the Society for SAN, Grace Schenatto and Anderson Herculano from SBNeC, the Brazilian Society, Cristian González y José Valdés de la Sociedad Chilena de Neurosciences, Gloria Patricia Cardona y Zulma Dueñas from the Colegio Colombiano de Neurosciences, Giselle Prunel and Francesco Matias Rossi from the La Society from Uruguay. Thank you very much. Without all of you, it would be impossible to have this meeting. The last, I wanted to remind you, the president of the Brazilian Society is going to talk about it a little bit, of the third FALAN Congress that is going to occur next year in Belém. Belém is very close to the Amazon River. This is the dolphin, the pink one that we can find in this place. This guy, the Colombians maybe knows, it is Walter Stuber. It was in a VLTP course that I organized in Manaus. And we worked very hard, but we could see these wonderful things in the river. And maybe you feel attracted a little bit more for not just by science, a lot of by science, but to see what happens in that region. That is a really wonderful region. And the last thing, you can see here our committee. I think I have a friend now in Chile, Colombia, Uruguay, Argentina. Thank you very much. It was, I feel sorry that we are leaving each other, but we will meet, I'm sure about that. And I would like to say thank you also to Danielle and Meli and Ismael. I think all the members of his company, of his team, that really helped us a lot and made possible this wonderful meeting.
It is an honor to be here talking to all of you. And my mission, it's a mission here, is to give you an update on our plans for next FALAN meeting in Brazil. As Elaine already mentioned, the meeting will be in Belém do Pará. This is in the north of the country, close to the Amazon forest, the Amazon River. It's in the same region. And the dates for the next FALAN meeting in 2022 will be from 11 to 14 of September. I feel that I need to give you some explanation about what happened. Of course, we had worked relentlessly to organize a meeting in 2020, 2 years ago, but we were caught up in this terrible pandemics of COVID-19, and we have to postpone the meeting. The idea was to postpone to 2021, but again, because the conditions, the sanitary conditions weren't ideal, we postponed it again to 2022. Now, I believe that we will be able to have this meeting, this most needed meeting, especially after this one, which was a virtual one, but very successful. And it will be so rewarding to have everybody together on a face-to-face meeting. That's our goal. However, if guests, some of the guests do not feel confident or comfortable to come, we are planning and we are working on a hybrid meeting. Hopefully, it will be a face-to-face, but we have to be prepared for any obstacle that may come in our way. The good news is that some of the guests and keynote speakers already confirmed the participation in this meeting. So I believe that we will have a wonderful and very exciting program that will appease to everybody working on the different areas of neuroscience. We also are mapping all the original proposals, if they are confirmed or not. And if we feel that there are some slots, we will launch new callings for proposals. So stay tuned on the meeting site, because we will reach out to you asking for new proposals and new poster submissions. Everything will be reopened so you can participate in this meeting. And finally, I would like to assure you that during these 2 years, the organizing committee didn't stop working because our goal is to bring the best meeting possible. So we have met and we are still meeting regularly and working relentlessly to make a great meeting of neuroscientists, not only a meeting, but a reunion actually. So we can come together and exchange ideas next year. So thank you very much for your attention, and I hope to see you all next year in Belém do Pará.
Você tem certeza que conhece o mundo ao seu redor? Amazônia, 5.500.000 quilômetros quadrados. Uma complexa e ainda desconhecida biodiversidade regida pelas águas que equilibra a vida do planeta. Para se conhecer os segredos da Amazônia, a porta de entrada é Belém, a capital do Pará. O estado que melhor define o verdadeiro significado da palavra Amazônia.
Okay, Patricia, Gloria, Paola, please, if you would like to say something. Gloria is going to be the only one of us that is going to keep working, so good luck. And if you need us, we are here, okay? And Patricia and Paola. Thank you very much, all of you. We are really, really happy. Patricia and Paola, Patricia, you with your enthusiasm, and Paola with your method. Great. I'm very happy. Thank you.
I would like to say something. Thank you. For me, it was an honor to share with you. Really was enriched of a lot of experience, a lot of learnings. I will continue in the committee. We will try to work strong with Deborah and all the people in the committee from the Society of Brazil and of Neuroscience and Behavior, working for the next meeting and trying to discuss a lot of these politics about how improve the neuroscience in Latin America. Thank you, Patricia. Thank you, Paola, for your valuable Misha's valuable teachings and for all your enthusiasm in this preparation of this meeting. Thank you to all the organizing committee from the different representatives of the Society of Neuroscience in Latin America. Thank you, Deborah, for all your enthusiasm also to continue with the presential meeting the next year. Thank you very much for your presence. Thank you to Daniel, to all the technicians for support and to be possible this meeting. Thank you very much.
Thank you to Belen, that's not here with us today, huh?
Bye.
Okay.
Thank you.
Okay, thank you very much again. And see you next year, September 2022 in Brazil, huh? Belém do Pará, all you. Bye-bye, bye-bye.